Okay, I think it's time to start for us. It's 10 o'clock, actually it's 1 after 10. And I would like to welcome you all to the session 1A. And our first presenter will be Valentina Catanzaro. Oh my gosh, I'm sorry, Catanzaro. Sorry for wrongly pronouncing your surname. And the title of the presentation will be COVID-19 lockdown impact on columnar and surface aerosol properties over southeastern Italy. Would you like to start your presentation now, or would you like me to share your presentation first?
Good morning to everyone. Now I'm going to share my video and I don't know what is going to happen with my laptop, but I can't see anything.
Okay.
Can you hear me?
Yes, we can. Can you hear us?
Yes, I can hear you, but
I
don't know what is going to happen. Maybe we can use your PDF and I can go on.
Sure.
Okay, thank you very much. I'm sorry for this problem.
No, it's okay.
What is going to happen? Okay, can I start?
Let me share your presentation first.
Okay.
Okay. Can you see your presentation now?
Okay, I don't see nothing, but I can do.
Well, is everyone else seeing the presentation I'm sharing?
Okay, so I can start.
I can see the presentation.
I can see.
Okay, that's
Okay.
cool. And let me know when
the presentation.
Okay, so I'm going to start.
Yes, and let me know when you want me to change the slide.
Okay. Oh, okay, I'm here. I'm here again, but I don't know what is going to happen. So this is the first slide. In this first slide, I'm going to present my work that deals with lockdown 19 COVID impact on air quality over the south of Eastern Italy. You can go to the second slide, please. In this study, we have investigated the effect of lockdown 19 pandemic restrictive measures on the columnar and surface air quality in Apulia, which is a region in the southeast of Italy. We measured, we have also investigated the change of aerosol optical parameters in particular, the optical aerosol depth using the AeroNet photometer measurement that is operating in Lecce, and the changes in mass concentration of the PM10 and the PM2.5 and other main polluting gases from different monitoring sites of the Apulia Region Air Quality Agency. You can go to slide number 3, please. So here we have a definition of the aerosol. Aerosol is a suspension of solid particles or liquid droplets in air or other gas. Aerosols, as you can see in the pictures on the right side, can be natural or anthropogenic. For example, natural aerosols are dust, desert dust, pollen, or marine spray. And also we have anthropogenic ones that derive from the combustion of fossil fuels, particles produced by vehicular traffic, and also smoke. And then we have the main particles that we have analyzed are PM10 and PM2.5 that have an aerodynamic diameters typically less than 10 nanometers and 2.5 nanometers respectively. And they are comparable with human air or a fine beach sand. Next slide, please. Okay, here you can see that we have used two different devices. The first one on the right, on the left is ARPA Puglia monitoring air quality samples, and then the AeroMet photometer. Thanks to ARPA stations, we have analyzed 9 different stations, sites, including PM and gases measurement, while the AeroNet photometers belong to a famous global network, the so-called AeroNet, and it can be used to determine the columnar properties of the atmospheric aerosols. Okay, next slide, please. Okay, here we have that around December 2019, COVID-19 acute respiratory disease from the SARS-CoV-2 was discovered in Wuhan in China, and then it arrived in Italy in February 2020, when the first confirmed case of COVID was declared. Then the Italian government on the 8th of March 2020 declared the lockdown restrictive measures, which allowed people to go out only for work and for health issues. Next, please. Okay, here in order to investigate the effects of the restrictive measure adopted by the government, we have firstly studied the changes in a mass concentration of PM10 and PM2.5 in Campi Salentina, which is a town very close to Lecce City. Here we have that the daily mean values parameter of the parameters have been analyzed in 3 different periods. We have considered the pre-lockdown period, the during lockdown period, and the post-lockdown period, and the temporal intervals are reported there in the slide. So every period consists in 69 days. The main result in this first slide is a significant reduction in mass concentration of the parameters investigated in the period after the lockdown. You can go on, please. Okay, here on the right side of the slide, I report the box and whiskers plot corresponding to the data shown in the previous slide. A significant reduction of PM10 and PM2.5 mass concentration is here visible in the post-lockdown box, which I indicate with the red arrows. And mass concentration of PM10 after the period of lockdown is 50% lower than the mass concentration before the lockdown. And the same is for PM2.5, which have a decrease of 65% in mass concentration after the closing period. Okay, next slide, please. Then we have use a statistical analysis of the daily mean values of data for the 3 periods using non-parametric tests, such as the Kruskal-Wallis test that then was followed by the Duns test, and in order to understand if there were significant differences between the period of examination. And the result of the Duns test that compares each couple of periods has demonstrated that the periods are all different from each others. Okay, next, please. Okay, here in this slide, we have made a comparison between the reference profile, which refers to the average daily mass concentration of the previous 3 years, so 2017, 2018, and 2019, and we have compared this with 2020. Here we can see that the white box represents the months of the reference profile, while the gray ones are the months of the 2020. Then this comparison has shown that the effect of the lockdown measures on a PM10 mass concentration is visible in April, and I indicated with the red arrow, and then we can observe a delayed effect in June, in July, and also in October. Okay, you can go on. Okay, in this slide I reported also the evaluation of the evolution of the APD, that is the average percent departure, calculated every 15 days. Regarding the PM10, there are many significant reductions indicated by the green arrows, and the largest decrease was observed in April. And then there were also a significant reduction in June and July. You can go on. Okay, here with the red line, we have the PM2.5, and also for PM2.5, we observe a very similar temporal evolution, but the decrease of APD was more relevant because generally in our side, we have a larger contribution of the fine particles. In particular, here for PM2.5, we obtain a reduction of the 65% in June. Okay, you can go on. Okay, here we have the comparison between the APD from the ground-based PM10 collected with ARPA Puglia monitoring station and the one from the Arionet photometer based on the columnar air optical depth data. As you can see here, we have that the temporal evolution is very similar with a significant reduction in June, and also we found a good correlation if we are comparing, even if we are comparing ground data and the columnar data. Okay, you can go on. Okay. Here, from now, I have shown data from Campi Salentina, but as I say at the beginning, we have also analyzed other monitoring stations from the ARPA region air quality agency, such as Lecce Libertini, which is a station located in the center of the city, so it's a urban station, urban traffic. And then we have Cerrate, which is a background, also an industrial site like Surbo and Maglie, which is a very small town. Here we have that PM10 and PM2.5 mass concentration in these areas have the same trend of Campi Salentina's data analyzed. And also here there is a reduction in mass concentration of the parameters from May until October. Then you can go to the last slide. Okay, here. So in conclusion, we have observed that thanks to the restrictive measures caused by COVID pandemic, PM10 and PM2.5 mass concentration decreased after the period of lockdown. So we can say that there is a delayed effect. Also, we have that a mean decrease of PM10 and PM2.5 is equal to 50% and 65% respectively. And also we can say that the APD decrease trend of the columnar AOD restrictive from some photometer measurements was very similar to the one of the PM10. Okay, I am finished and I'm going to thank you for the attention.
Okay, thank you very much for your presentation. I think we have 1 minute left for some questions from you guys. And if there are any more questions later, just put them on the chat and talk. Okay, any questions for Valentina?
I'm Luca Ferrero. I have a question, a curiosity.
Yes.
Because in the North Italy, we have seen some change in chemistry, but not in the mass of the particulate matter due to the lockdown, probably because the Po Valley is a huge emitter, so we cannot catch the mass change. But in this respect, are you going to investigate also the retrieved parameter of aerosol, like the size distribution, or the refractive index, maybe using the Arionet data in order to understand if any change in the type of aerosol?
Yes, thank you for the question. This is the first step of our analysis. So we are going to continue the analysis using other monitoring stations from ARPA Puglia and also including more analysis from the photometer, because we wanted to analyze the data from the ground and the air column. So Arionet is a very important devices for our work.
Okay, is that okay? So can we continue with our session 1A? To the next person, and Maria Pia Romano, please take your voice and on electrochemical DTT detection to assess the oxidativa potential of PM. Would you like me to share your presentation, or will you do it by yourself?
Good morning to everyone. I tried to share the presentation. Just 1 second. Is okay? You are able to see?
It's still loading, but yes, right now we can see it. It's perfect.
Okay. Okay. Well, I prepared a presentation for people who haven't an extensive knowledge in this field. So just, I'm not able to see there is a problem. Oops. Okay.
Can you see your, yeah, we can see your presentation.
For 1 second, the monitor was black, totally black.
Okay,
I don't know. Okay.
It's fine.
The presentation is for people who haven't an extensive knowledge in this field. I'm going to speak about the topic of my innovative PhD. The title is an innovative electrochemical sensor for tiles detection, a device for the measurement of the oxidative potential of particulate matter. And I realized this work in the last year, and with a big research group. These are my tutor, Maria Giulia Leonetto, Maria Rachele Guascito, and we work together. Okay, it's impossible to go ahead. There is a problem.
You need to slide it down.
Okay. What is PM? Like Valentina, I chose this image because it's particularly significant in my mind. You can appreciate the very small dimension of PM. The term particulate, in fact, refers to the solid and liquid particle dispersed in the atmosphere, and is the major responsible for the oxidative stress. The exposure to the particulate matter has detrimental effect on our health. It's very well known. You can see in this image a human hair and a fine sand beach. You can appreciate the dimension of PM10 and PM 2.5. What is the DDT? We are going to speak about DDT. The dithiotrietol, also known as Cleveland reagent, is a thiol, and they are the molecule with a typical smell of rotten eggs, but they are very important because they are involved in biological process and also environmental process and phenomenon. The electrochemical sensors are useful in many fields connected with human life, and for example, environmental analysis, and also they are useful for monitoring the air quality. So go on. The aim of this study, we want to focus our study on PM toxicity and cytotoxicity, and on its oxidative potential. In fact, this sensor can be considered a device for the measurement of the oxidative potential of DOP. And we want to investigate the mechanism by which PM impact human health. The hypothesis at the moment is that PM is responsible of damage of human health by producing reactive oxygen species, the ROS. In this image, you can see the different dimension between PM10 and PM 2.5. This study is an interdisciplinary study because on one side we do the electrochemical test, DDT, a cellular test, and you can see a typical voltammetric curve or amperometric detection, and on the other side we do biological assay on a specific cell line, A549, just to test the damage damage caused by PM on human cells, because this type of cells are cells derived for human lung. The study was carried out on PM water soluble fraction because the PM was collected and then extracted. We have to say thanks to Daniele Contini teams of ISYC CNR of Lecce. There is a long-standing partnership with the University of Salento, in particular with Disteba. So the innovative sensor, we can say, is innovative because this sensor can be considered an alternative to the classic spectrophotometric method, and the operating principle is the determination of the oxidative potential through the measurement of the loss in DDT. In parallel, we do cytotoxicity and induction of oxidative stress by MTT and fluorescent probe. So on one side, the electrochemical test, just to validate the sensor, and on the other side, the biological assay. Let's have a look to the health effect of the exposure to PM. It's very well known that airborne PM induce harm with the generation of ROS. We have to make a distinguish between long-term exposure and acute exposure. Long-term can cause a risk of cancer, for example, and acute exposure can cause DNA damage and mutation. And air pollution is detrimental for human health. In particular, can penetrate into the lungs, and lungs are the first site of exposure. For this reason, concerning the cytotoxicity analysis, we have studied the A549 cells, which is a cell line, immortalized cell line derived from human lungs. The cytotoxicity and the induction of oxidative stress was evaluated by MTT-assay on one side and with the fluorescent probe on the other side. The cell vitality test is a DMTT test. It measures the activity of one enzyme that catalyzes the reduction of DMTT bromide. DMTT bromide is usually a yellow fish salt. Thanks to this test, it changed the color in a formazan which has a purple colors. In this graphic, we can see the mortality for the sample. We analyzed, for example, 10 samples. For the sample number 8, the mortality was very high, about 60%. In this image, we can also see the dead cells. The MTT show an increase in cell mortality after only 24 hours of exposure. Concerning the cytotoxic effect of PM, we use the fluorescent probe. I don't want to go particularly inside, but I have to highlight that the fluorescent probe is very important because it generates an oxidation and a fluorescent adduction that is trapped inside the cell, and we can study the cell for a long period. So we can appreciate, for example, the apoptotic bleb. There is a typical cellular damage. The cell membrane appears like that with no defined lines. And the exposure to PM extract is highlighted in this image, and the result in a production of ROS, reactive oxygen species. We do also a correlation, and we can a correlation between MTT analysis and the fluorescent probe, and we can say that the higher the intracellular oxidative stress, the higher the mortality observed. Concerning the dose response, the higher the PM concentration, the greater is the stress induction on the cells. Let's have a look now to the electrochemical part of the work, that is the acellular test. In this picture, you can see our laboratory in Lecce. All the electrochemical experiments in batch have been carried out with a drop sense electrochemical workstation. You can see also the conventional 3-electrode system we used.
Maria, I'm sorry, can I ask you to sum up your presentation so we have time for the questions for you?
Okay. Let's look to the electrode preparation. We do a modification on a glassy carbon electrode and with a gold nanoparticle. Very simple, the preparation of the electrode. The first step was drop casting. The second step was a chronoperimetry to perform the electrode deposition of gold. And the third was the cyclic voltammetry to stabilize the system. Concerning the sensor characterization, we study through cyclic voltammetry and the chronoperimetry. You can see in particular here you can see the electroactivity, and in this picture you can see the difference between the glassy carbon bare and our sensor, which is modified with gold nanoparticles. We get a lot in terms of current, and you can appreciate also the gold nanoparticle. They are randomly distributed on the glassy carbon, and they are a spheric shape, and the average diameter is 20-50 nanometers. Okay, we do also the experiment in flow injection analysis, and we obtained encouraging results also with the FIA analysis. Conclusion: the biological study with MTT assay and fluorescent probe show the intracellular oxidative stress induction by PM, which in turn result in increased cytotoxicity. Concerning the electrochemical study, we can say that the sensor is a good sensor, is an innovative sensor. It operates well, it's reactive towards DTT, but not towards the other interference such as PM. And in the trend of sensitive detection, the electrochemical sensor promotes simplicity and high sensitivity with a minimal equipment. So thank you for the attention, and sorry for the delay.
No, it's perfect. And now we have time for, I think, one question, and the rest please write on the chat. So Maria can respond to you.
I'm not able to see the chat because I stopped the. Okay, stop the in chat. There's no question for me.
Yeah, I know. This is for later if we have no time. Okay. Are there any quick questions for Maria for now?
No question for me.
Okay, if you have any questions for Maria later, please write your question via chat so we can read it and Maria can respond to you. And our next presenter will be Katarzyna Sindera with fish gills as an inspiration to create new effective catalytic carriers. Okay, would you like me to share your presentation or?
Hello, everyone. I think that I will try to do it by myself.
Okay.
Okay, let's see if I'm able to. Okay, can you see the presentation?
Yes, just put on the full mode of presentation.
Okay.
So, because now we can see the comment section. Oh, yeah, now it's working.
Is it okay now?
Yes.
Okay, my name is Katarzyna Sindera. I work in the Institute of Chemical Engineering of the Polish Academy of Sciences. In my presentation, I will try to ask the question of how fish gills can contribute to the development of modern catalytic carriers. I will start by describing the problem faced by chemists in the field of catalysis and chemical engineering, but of course not only. Over last decades, there have been a rapid development of the industry, and as a result, the increased amount of dangerous substances has been produced. These substances are methane, dangerous greenhouse gas, nitrogen and sulfur oxides involved in formation of acid rain, particulate matter, as Maria Pia Romano said before, very dangerous for our health and environment, and wide group of volatile organic compounds. A significant portion of VOC is poisonous, carcinogenic, and mutagenic. It has been estimated that around 3.22 million people died globally as a result of diseases caused by air pollution in 2010 alone. So these numbers are very scary for us. Catalytic processes enable effective elimination of toxic emission. In environmental application, heterogeneous catalysis is of great importance. For example, catalytic combustion and NOx abatement are integrated in catalytic car converter. Catalytic filling consists of catalytic active phase and support. Oh, that's better. Catalytic active phase consists of very expensive noble metals. This is very important part of the catalytic filling because it is important for the course of chemical reaction. But if we will be able to make all the reactor filling only for example, platinum, it would be so expensive that its large-scale production will be totally unprofitable. This is the reason why catalytic supports are used. On the surface of catalytic support, only thin layer of expensive catalytic active phase can be deposited. This is the reason why the shape and the geometry of catalytic support is very important. The most commonly used in industry in large-scale applications, catalytic supports are monoliths and packed beds. The perfect catalytic support should ensure intensive heat and mass transport between flowing reactants and catalytic active centers, large specific surface area, small reactor dimensions, it's important especially in mobile applications, and reasonable flow resistance. For me, as a constructor, the most important thing is the intense heat and mass transfer properties of the catalytic support. Aim of the study is to develop a new type of catalytic supports, the modified geometry of which would ensure easy access to the active centers deposited on it. It is possible to create such a geometry that would provide low external diffusion resistance, intensive heat and mass transport, and reasonable flow resistance. Nature, thanks to the evolution, improved the process of heat and mass transfer during much longer time than any human would be able to. It has been experimentally proven that 80 to 90% of heat generated in the fish organism is exchanged as it passes through the gills. That means that nature constructed very efficient heat and mass transfer exchanger, and perhaps maybe if we construct a new catalytic carrier on the dimensions based on the shape of fish gills, we will be able to construct the efficient catalytic carrier, maybe as efficient as fish gills are. On the slide, there is presented the morphology of fish gills. The most important part are filaments and lamellae. Can you see the mouse? My mouse moving?
Yes.
Okay, so this is the diagram, and there are pictures, some pictures of natural fish gills of two different species. This is the gills of snow trout, and these are the gills of Florida pompano fish. The filaments have elongated shape. You can see it here, there, and there. And the lamella are mounted on its surface. The lamella have plate-like shape. And what is important and surprising, for different types of fish, there are different dimensions of these morphotic elements and different filament length to lamella length ratio. So the question arises, what should the dimensions have the efficient catalytic carrier created by engineers? This is proposed geometry of the carrier. It contains the above mentioned elements like filament and lamella. This is the side view, and this is the top view. What is important here, and for me very sad, is that the techniques that enable to produce that type of construction do not allow to produce it in so small scale as natural gills have. So in case of natural gills, we have nanoscale, and in case of my gills, we have millimeter scale. I conduct my research with the use of CFD, computational fluid dynamics. It is a very useful tool used many times by my team. Such examples are presented on the slide. What is very important for us is that the results for other types of carriers were in sufficient agreement with the experimental ones. CFD consists of 3 steps. The first is pre-processing, preparation of geometry, and mesh for simulation. What's important, I didn't prepare the geometry of the whole carrier, but only of the repetitive unit. I prepared CAD model in ANSYS Design Modeler module, and there is the mesh presented prepared in ANSYS Meshing module. Of course, meshing is dividing the whole domain into small elements of finite volume. In this small finite element volume, calculations are conducted. Mesh was consisted of tetrahedrons in Gills part and hexahedral elements in the most part of control regions. Control regions are here and here. Mesh consisted of about 9 million elements, and the mesh was transported into polyhedral type using Fluent module to reduce CPU usage and simulation time. Of course, there are some important parameters for checking mesh quality. And then I conducted simulations and I resolved the analysis. These are analysis of my CFD simulations. All these pictures are for the case of inlet flow velocity of 0.1 meter per second. These are the temperature distribution in the carrier. This is the same carrier, but seen from different axis perspectives. This is for X axis, I mean from the top, Z axis from the back, and X axis from the bottom. And there are also particle distribution in the carrier. What is very important for me is that behind the carrier, there are no great swirlness that could bring some pressure drop, larger pressure drop. Okay, these are the results. There are transport flow characteristic. We've got characteristic for heat transfer. Nusselt number versus Reynolds number, and the characteristic for pressure drop, it's the pressure loss per length of the catalytic filling versus superficial fluid velocity. And the characteristic for Mark carriers are compared with the characteristic for packed bed and for monolith, which, as I mentioned before, are typically used in large-scale applications. As we can see, Gill-inspired catalytic carrier have larger heat transfer properties than monolith, but not as high as packed bed. In case of pressure loss, this pressure loss generated by my carriers are much lower than in case of packed bed, closer or even equal to monolith. And conclusion, Gill-like structures have flow resistance equal or higher than the monolith, while at the same time much more intense heat transfer than the monolith. The examined structures generate much lower flow resistance than packed bed, although the transport properties of the tested structures are much lower. Gill-like structures required further research to determine what dimensions of characteristic morphotic elements are key and most beneficial for the best transport flow properties. And that are my studies for future. Okay, thank you very much for your attention.
Thank you very much for your presentation. And now, are there any questions for Katarzyna?
Okay, stop share. Oh, I've got nice time.
Yes. Hello, hello. Are there any questions?
Are there any questions?
I have, if I may. Can you give an example of such structures that have been already done in real life? I don't know, a structure of a bridge, building, something?
But I'm not sure if I understand correctly. You mean the examples of catalytic carriers, the catalytic structures, or my one, fish, there's gill-like catalytic carriers?
Because I understand that the structure can be used pretty much everywhere, right? Because,
okay.
So are there any real life examples of using it, or?
Of course, when we are talking about catalytic carriers whole, there is one surprising application for non-chemists.
That's exactly my question.
Yes, yes, that I want to mention. You know, every car needs to have a catalytic converter. We call it commonly a katalyzator samochodowy. Okay, and there the monolithic structure are used very, very commonly. Okay. And this is because you can, you know, in your car, you've got two working reactors. One is the engine, and the next one is the catalytic car converter that I mentioned. And you've got there the catalytic support, and maybe someday in the future, it will be my gill-like catalytic support. Now, the most commonly used is the monolith, and on the surface of which is the layer of platinum applied. That is the reason why the catalytic converters are commonly steeled, because the platinum is so expensive. Sure. The most important task for this catalytic converter is to clean the exhaust gases from the engine.
Okay, thank you.
Okay, that's very interesting. I am sorry to interrupt, but we have to continue with our next presentation from Beatrice Sorrentino, have the green groups an impact on the atmosphere?
Okay, thank you very much.
Thanks.
Good morning, can you hear me?
Yes, we can.
Okay.
Would you like
to yes, I try to, one moment, yes. To share the screen. Can you see the screen?
It's loading? Yes, we can see it.
Okay, just full screen or yes?
Yes, right now.
Okay. So, good morning. My name is Beatrice Sorrentino. and I am a temporary researcher at the ENEA agency, which is the National Agency for New Technologies, Energy and Sustainable Economic Development. I am a nuclear physicist, but now I am working in the production and the processing of meteorological and air quality data in the framework of Live Gap project, which is the vegetation for urban green air quality plans, in which I work with my tutor, Michaela Mircea. And today I want to give a brief overview about the project and give some details about objectives. So let's start. We need to start with one of the greatest contemporary challenges, which is coping with the global warming. I believe that to tackle with this problem is necessary to have an overview that allows us to define plans to face it. To better understand what I mean, let's make an example. The problem in its overall view is the greenhouse effect and consequently the global warming. And one of the causes is, for example, deforestation or the intensive farming. So what I want to say is that dividing a problem into sub-problems is very important because it makes it easier to establish which are the most suitable methods to tackle it, but also because even the smallest aspects can make a difference. Now, let's us consider the problem from at the city level. Changing the structure of the Earth's surface lead to a phenomenon called urban heat island. This term indicates a metropolitan area that due to human activities has a higher temperature than the surrounding areas. In this slide, you can see some causes that increase the temperature. As you can see, anthropogenic emissions or the lack of green areas in our cities. To mitigate the urban heat island phenomenon, nature-based solutions can be used. The management and sustainable use of nature, such as the construction of green walls and or green roofs in cities or urban greening. I think that the use of a sustainable approach to mitigate climate change is more fundamental than ever. In details, now I am studying the impact of green roofs on the atmosphere in the city of Milan. Because contrary to what one might think, vegetations not only bring benefits on air quality, but also the reduction of anthropogenic emissions, but also emits in a very different ways from species to species, biogenical volatile organic compounds called BVOCs, such as isoprene or monoterpenes. These compounds react in the atmosphere with nitrogen oxides to form ozone, which is a very toxic pollutant for humans. For this reason, the main goal of the project is to understand the effect of the urban ecosystem on air temperature and evaluate the impacts on air quality with regard to the main pollutants. In particular, as I said before, I am working with the green roofs in the city of Milan in Italy. And here in the slide, you can see the domain, which is formed by a 51 per 51 kilometers grid. And the black outline is the municipality of Milan. This study using geographic information system, which involves information produced by Milano Geoportale. As shown in this figure, in this map, you can see the distribution of the buildings in Milan, in the city of Milan. And from this data, the percentages of green roofs already present in the city, but also the potential green roof, which means those that can be built in the future, are extracted. But I cannot show you the map due to the copyright. But to better understand what I mean with the increase of green roof in the city of Milan, I made a zoom on the map, and in the map on the top, in dark green circles, you can see existing green roof in the city of Milan, and at the bottom, in green light circles, you can see the potential green roofs. Numerically, today, there is a coverage around 2.5%, which will become close to 32%. At this point, considering the potential green roofs, geographic information were integrated with the most suitable green population for green roofs. Several factors contribute to the optimal settlement of vegetation. For example, climate factors such as humidity, rainfall, or exposure. Many rules governing plant selection in green roofs, and they discard the vast majority of annuals and perennials plants, and including also several native plants. In this case, xerophilous species have been chosen because these species easily adapt to the environment and climate. And among these species, we can find Sedum acre, also known as gold moss stonecrop, or Dianthus, which is known as carnation, but also thyme, rosemary, santolina, and potentilla. After the chosen distribution for this species is 70%, but except for the Dianthus, which is 15%, as you can see in this table. These choices came from the collaboration with Silvano Fares from CNR, which is the National Research Council, and Ilaria Zappitelli from CREA, which is the Council for Agricultural Research and Economics. After this, data were transformed from vector to raster data in NetCDF format for the modeling system. After, the polygons of the scenario were normalized, and the percentage of land occupation is calculated. Here, the example shows the distribution of Sedum acre on the left and Dianthus on the right through the whole municipal area. From the legend, you can see that the largest cell with the Sedum acre fraction is 0.0275 square meters, which means that this cell have an increase of Sedum acre on 27,500 square meters, as the total area of the cell is 1 to 6 square meters. And here, the collaboration with Giuseppe Cremona from ENEA was fundamental. At this point, the input for the modeling system is prepared. The modeling system, called WRF, has a meteorological model, the weather for research and forecasting model, which is a next generation mesoscale numerical weather prediction system designed for both atmospheric research and operational forecasting simulations. But also, after, natural emissions are computed with SURFPRO model, and eventually the chemical transport model called FARM, which is flexible air quality regional model, estimates concentration of the main pollutants as PM10. So at the end, once the simulation for the proposed scenario have been obtained, results are analyzed to integrate new information to better design the urban air quality plans. But another useful goal of the project is to provide recommendation for improving the legislation aimed to improve air quality in European cities. For me, that's enough. I'm finished, and thanks for the attention.
Thank you very much. Now we have time for questions. Are there any questions for Beatrice?
I interrupted.
Yes, I've got a question. It's Valentina.
Hi.
Okay.
Hi. I want to know if you are considering to use other green infrastructure, for example, to reduce the thermal heat on the city and to improve the outdoor thermal comfort.
Yes, yes. Also, green walls. But I'm talking about the green roof scenario. But we're studying also other scenarios like, for example, actual vegetation, future vegetation in the city of Milan, but also in the city of Bologna and Madrid. And or another scenario, another developing scenario is the private green scenario. And so, yes, we have many other scenarios to
Thank
study.
you. I ask you this question because here in Lecce, we did something very similar by planting other trees and for evaluate the thermal comfort, the outdoor thermal comfort, and we gave some I don't remember the name. We take some interview to people to understand if their green infrastructure can help in the air quality.
Okay. We also consider other parameters such as albedo and also the reflectance of buildings, and we taking into account many other things, not only vegetation.
Okay, thank you.
Thank you very much.
Okay, are there any other questions? If you have later on any questions, just put them on a chat or send it to us and we will direct it to Beatrice or any of you.
No, nothing.
A quick question. I'm Luca Ferrero from Italy.
Hello?
Very quick question. Did you perform some preliminary run on the?
Yes, we have some preliminary runs, but yes, there are many. The first run I did 2 weeks ago is changing the albedo parameter, but it's we also study this. We have no definitive results, so we're not sure. For now, we're not sure. But we have to better understand. We have to better understand what we have from the simulation. But yes, we did some simulations, some preliminary simulations.
Okay, because as I work in Milan also with the vertical profile measurements of pollutants and so on, it should be very interesting if you are maybe to compare in the future.
Yes.
Okay.
Of course. Thank you.
Okay, any other questions? Okay, if not, then we will end this session 1A, and later on we will meet 11:10, either on this session still, or if you would like to switch for session B, I put the links at the beginning of this session, so you can follow them and choose whatever you would like to hear. And this was really nice to hear all of your presentations, and thank you very much for your time and really nice projects. Okay.
Thank you very much. Fantastic timing.
Good morning.
Good morning. Hi, Sarah.
Hi.
Good to see you.
Nice to meet you.
Nice to meet you too.
If you want, I can try to share my screen.
That would be perfect. Yeah, we will earn some minutes to do that.
Okay. So So it's not already possible for me.
Okay, so probably we need to
You
ask.
have to stop with your condivision.
Yeah, right now you can try.
Okay.
Thanks, Ola.
Good morning.
Morning.
Hello, Szymon.
Okay.
Hi, hi.
Now I think I have to. Go on my presentation. Okay. Okay.
Perfect. So it's 11:10. So I think that we can start. Welcome back after the break. My name is Szymon Smoliński. I work for the National Marine Fisheries Research Institute in Gdynia and the Institute of Marine Research in Bergen. And I will be chairing this session. And in this session, we have four talks. All of them are related to biology, but they cover different groups of organisms. So I'm really excited. And I have just very short reminder for the speakers that you have 12 minutes for the presentation, and then we will have 3 minutes of questions and answers. So I'm a rather strict chairman, and I will let you know after 10 minutes and then stop you after 12, just to go for these questions. And now I would like to introduce our first speaker, Sara Caroni from the University of Milano Bicocca. So yes. Sara, the virtual floor is yours now.
Yeah, thank you very much. Good morning to everyone. Today I'm going to present you the first results of an extensive research project that focuses mainly on some particular and peculiar traits of the biology and on the ecology of the most abundant species of macroalgae belonging to the genus Caulerpa that are currently present in the Mediterranean Sea. The study was and is already conducted in collaboration with the University of Sassari and the Marine Protected Area of Tavolara Punta Codacavallo along the northeastern Sardinian coast. Today, in particular, I'm going to focus on the effects of interspecific competition on the main morphological, cytological, and physiological traits of two species belonging to the genus Caulerpa, Caulerpa cylindracea and Caulerpa prolifera. The first one, Caulerpa cylindracea, is an alien species particularly invasive in the Mediterranean Sea, where it was recorded for the first time in 1990. And as you can see in the image, in less than 30 years, colonized all the basin, and it today is particularly abundant in all the Mediterranean Sea. Caulerpa cylindracea is able to settle on different types of substrata and to withstand competition with both seagrasses, autochthonous seagrasses like Posidonia oceanica, and autochthonous macroalgae. And where Caulerpa cylindracea is present on the substratum usually presents a reduction in the structural diversity that usually characterizes the substrata of the Mediterranean Sea, and usually also a significant biodiversity loss occurs in such case. Caulerpa cylindracea competes very actively in the Mediterranean Sea with Caulerpa prolifera, another species of the same genus that is native of the Mediterranean. So the two species are congeneric species, and for this reason they are expected to have the same morphological, cytological, and physiological basic scheme. But it is also well known that invasive alien algae usually have superior competitive abilities if compared with those of autochthonous species, and thus because they have usually some peculiar morphological, cytological, and physiological specific traits that are useful to this competitive, to increase these competitive abilities. Moreover, in the last few years, some studies underlined how seaweed can undergo significant changes in the morphological, but also cytological and physiological main features in relation to competition. So considering that the two species, as I was saying before, compete very actively in the Mediterranean Sea, and that no information on such competition is available in literature, it appears for us very interesting to try to investigate and highlight the main differences in the main traits, speaking about morphological, cytological, and physiological traits between the two species, also in relation to intra, but especially to interspecific competition between them. For this reason, a manipulative field experiment was performed in Tavolara Punta Codacavallo Marine Protected Area, as you can see in the map. In particular, 24 experimental units were prepared in the field on hard substrata on which the two species are naturally present. You can see an image of one of the experimental plots in the slide. And the abundance of the two species was manipulated in plots according to different treatments, to 10 different treatments that allowed us to investigate at the same time both intraspecific competition for the two species and interspecific competition between them. The abundance of the species was manipulated, transplanting some fragments of each of the two species in the quadrats. Each fragment was represented by a thallus of one of the two species with at least 10 blades. The experiment was conducted in the summer of 2019 from July to September, when the development on the substratum of the two species is maximum. And on the whole, 4 samplings were conducted. We used an integrated approach to obtain data on both the species, trying to consider different types of variables. Morphological, physiological, and cytological variables were considered. You can see them in this list. In particular, we considered the blade length and the blade area of the two species present in the quadrats. For the area of blades, we considered all the thallus together because, as you saw, I think, in the previous slide, the blades of the two species are quite different, and blades of Cylindracia in particular are smaller but more abundant. Then we considered also the density of blades and the percent cover of the substratum by the two species. We analyzed the chlorophyll and the carotenoid content of algae in the different plots, their photosynthetic activity, and finally, we investigated also the chloroplast and the nucleus size of algae in the plots. All the results we obtained were then treated using statistical analysis techniques. In particular, we used univariate statistical analysis techniques with the software GMAV5, and ANOVA and SNK tests were performed. Now, I start to briefly describe you the main results we obtained, considering that we had a lot of data, and so it's not possible for me to show all the results. Only the most interesting, in my opinion, results are presented in this presentation. So first of all, considering data obtained from control plots in which only 2 fragments of one of the two species were present, we can see that higher values for all the variables were obtained for Caulerpa Cylindracea. In the plots, you can see all the variables we considered. In red, the bars in red are referred to Caulerpa Cylindracea, while the bars in green to Caulerpa prolifera. And it is clearly visible that higher values were obtained for Cylindracea, considering all the variables. The only exception is represented by the nucleus size, as the mean area of nuclei was significantly higher for the autochthonous species. Some interesting results were obtained also considering interspecific competition. You can see in the plot some of the most interesting results. And it is particularly interesting, in my opinion, that some differences were observed in the response to competition for the different variables we considered. For the photosynthetic activity, the content of pigments and the size of chloroplasts in particular, higher values in case of interspecific competition were obtained for both the species. In this plot, for example, you can see what happened to the chloroplast area in case of competition. And bars referred to high competition levels are those on the right in the two plots, one plot for prolifera and one for Cylindrasea. In this case, also the statistical analysis confirmed the significance of the difference we observed. And a very similar situation was recorded also considering the total chlorophyll content for both the species, an increase related to increasing competition. We are speaking of interspecific competition because, on the contrary, no significant differences were observed when considering plots in which intraspecific competition was present. Changing the variables, another result. So if we consider blade length, blade density, and also the percent cover of the substratum by the two species, in case of interspecific competition, higher values were observed only for Caulerpa Cylindrasea. And a quite interesting situation, in my opinion, was observed with regard to blade length, that are data reported in the two plots that you can see. No differences for prolifera, some quite interesting differences for Cylindrasea, and an increased blade length only in plots in which the number of fragments of Caulerpa Cylindrasea was higher than that of Caulerpa prolifera. When the number of fragments of the two species was the same, or when Caulerpa prolifera was more abundant, a very different situation was recorded with quite lower values with regard to blade length, as you can see in the bars that are underlined.
Excuse me, Sarah, 2 minutes left.
Yes, also in this case, statistical analysis confirmed our observations. Another result that is quite interesting regards the size of nuclei. Once more, no differences with interspecific competition for prolifera, while a significant reduction for Caulerpa Cylindrasea. So, what about this data? First of all, I can see that some important cytological differences between the two species seems to occur. And these data are in accordance with those of other studies that recently analyzed the competition and defined the main features of alien and native species, congeneric species. Caulerpa Cylindrasea had bigger chloroplasts, while Caulerpa prolifera bigger nuclei. And this is typical of species that are particularly invasive because small genomes are usually characterized by the greater invasiveness. And moreover, species with big chloroplasts are characterized, of course, by other photosynthetic rates, and these higher rates can play a key role in determining the invasiveness of the species. In our study, also some important morphological and physiological differences were recorded, thus confirming the hypothesis that cytological differences also produce morphological and physiological heterogeneity. And a different response in particular was underlined for the two species in case of competition, with Caulerpa Cylindrasea that seems to have higher performances that can, of course, favor its adaptation and acclimatation and play a key role in determining its invasiveness. But another important and very significant data were obtained. This is that regarding the fact that in units in which the growth of Caulerpa prolifera was also allowed, so where fragments of Caulerpa prolifera were present, the growth of Cylindrasea was not so high as in plots in which the other algae was not present. And on the other hand, no differences in the substratum cover and in the blade main features for the native species were observed also in units in which also Cylindrasea was present. So we can conclude that also prolifera is able to resist to the invasion of Cylindrossia, and it also seems to be able to reduce, at least in part, the spread of this alloctonous species. Thank you for your attention. I've finished.
Thank you very much, Sarah. I have to say that I'm really impressed. Field experiments is what we very often miss in marine ecology, so that was really great to see your results. Do we have any questions?
Yes, it's not so easy to perform such a kind of experiments in the field when we're working in the sea. But I think it's really interesting to try to obtain data that are collected in the field, because they really are a photo of what happens in natural environments.
Exactly, exactly. Do we have any questions? Please raise your hand or use the chat box.
No questions.
If not, thank you very much, Sarah.
Thank you very much.
Now
See you later.
Now I would like to introduce our next speaker, Dominika Hetko from the University of Gdańsk.
Morning. Do you see my presentation?
Yes.
No.
You can see it. Can you?
Yeah.
Oh.
Yeah.
Good morning. My name is Dominika Hetko, and I am a PhD student in the Doctoral School of Natural Science in the University of Gdańsk. I will present to you presentation about impact of the hydrodynamic condition of the recent diatom flora in the Puck Lagoon. The aim of the study is the interpretation of the hydrodynamic condition in the Puck Lagoon based on diatom flora preserved in the recent sediments. And in order to achieve purpose, the following research tasks were carried out. 129 sediment samples were collected in the Puck Lagoon using Van Veen grab sampler in 2019. And the qualitative diatomological analysis was performed. And next, identified diatoms was grouping based their preference in relation to the salinity and trophic status. And the last task was interpretation the results of Diatoms Analysis. As I mentioned it before, from the outer Puck Bay, from the area where I collected 129 samples, collected using Van Veen grab sampler. And the collected material was subject to diatom analyzing performed by standard Butterby procedure, which consists in removing unnecessary substances using chemical reagent. And to remove our calcium carbonate, samples were treated 10% hydrochloric acid. The organic matter interfering with the identification of diatoms was removed by using perhydrol. And next, the permanent slides were removed, and I performed quantitative diatom analysis. A total of 160 species belonging to 57 genera were identified. According to identified taxa, dominant benthic species, but also 20 planktic species were identified, and these species were observed in the southern part of the Puck Lagoon. Based on diatom flora in the research area, were separated 3 regions where the dominant diatoms prefer another salinity. And the first area is related to the northern part of the Puck Lagoon. And in this area, we observed a higher frequency of marine species such as Opephora gittergrassi, Opephora horstiana, and Opephora mutabilis. Also in this area, we observed a high frequency of Ophiphora crumbaini, which are brackish species, and brackish Planotidium delicatorum. Also in this area, the increased frequency of Amphora pediculus, which are freshwater species, we are observed. The last two species, Planotidium delicatorum and Amphora pediculus, is a species which we can observe it in all Puck lagoon, but in this area they have a high frequency. The picture shows dominant brackish and marine species, such as Ophiphora mutabilis, Ophiphora gintergrassi, and Ophiphora crumbaini. The second area is located in the central part of the Puck lagoon. And in this area, the increasing frequency of brackish Catenula adherens and freshwater species such as Achnanthes lemermani and Amphora pediculus have a higher frequency. The last area is located in the southern part of the Puck lagoon, and in this area we also observed abundantly Catenula adherens and Planotidium delicatorum, but in this area their frequency decreased. And at the same time, we are observing a high frequency of planktic species Cyclotella choctavachana, which prefer brackish waters. And what is important that these species are called anthropogenic. The high frequency of Cyclotella choctavachana in this area is related to the near in the coastal zone we are observing urbanization area. And the pictures show the dominance in the area 2 and 3, marine Catenula adherens, anthropogenic Cyclotella choctavachana, and freshwater species Achnanthes lemermani. The Puck lagoon are a reservoir with a low salinity. The average salinity in the Puck lagoon is 7.3 PSO. Therefore, the whole area is characteristic by a higher frequency of freshwater species. But in the northern part of the Puck lagoon, we observed a high frequency of marine and brackish species. And this is related to the inflow of salt water from the outer Puck Bay along to the Hel Peninsula. In the central and southern part of the Puck is associated with inflow of river's water. However, the southern part of the Puck lagoon is distinguished by a regrowth percentage of brackish species, and this is also related to the inflow of water from the outer Puck Bay. In the terms of trophic preferences, indicates a dominant frequency, diatoms preferring high edaphic requirements, and these species are dominated in the all area. A decrease their frequency in the northern part of the Puck lagoon is related to the inflow of the waters from the outer Puck Bay, and in the same time where the eutrophentic percentage of eutrophentic species is decreased, the percentage of eumesotrophentic species is increased. And summary, a total of 149 species belonging to 47 genera were identified in the all Puck lagoon. In this area were dominant benthic taxa. In the ore area, I separated 3 regions where diatoms prefer another salinity, and that in the northern part of the Puck Lagoon, inflow of salter waters from the outer Puck Bay causes the high frequency of marine and brackish species. And in this area, dominant species with genera Opophora. In the central part of the study area, the inflow of river waters have a significant impact of salinity. And in this area, we are observing a high frequency of freshwater species. And the last area is located in the southern part of the Puck Lagoon. And this area is characterized by a high frequency of brackish species and their dominant anthropogenic taxa Cyclotella choctavachana. The results of Diatoms analysis indicate that the Puck Lagoon is a eutrophic reservoir. The diatoms flora dominant in the Puck Lagoon, preferring high edaphic requirements. And in this area, we are observed 2 important sources of nutrients and organic matter. And this is sewage farm from Swarzewo city and inflow of rivers water such as Reda, Putnica, and Gizdebka. Thank you for your attention.
Thank you very much, Dominika. Do we have any questions from the audience? If not, I will ask a very short question. You showed us clearly that salinity is one of the driving factors of community structures. So I was wondering, because you showed us this influence of the salty waters in the northern part and fresh water close to the rivers. So I was wondering if it is possible, for example, to use diatoms to indicate, for example, artificial pollutions from highly saline waters, which is the case also for the Puck Lagoon. We observe this kind of situation.
Could you repeat the last words? I don't hear you.
I'm just asking, what is your opinion on the indication of the environmental status using diatoms? Do you think that diatoms can be used, to indicate, for example, discharge of highly saline waters?
Yes, I think the diatoms are cosmopolitans, microalgae, and in us research we using diatoms which prefer
I guess that the answer is yes, that it probably
yes.
You can use them. Yeah, that's interesting. I think that it is really labor intensive and you need to spend a lot of time analyzing samples, but definitely it can give you a nice overview of the environmental status. So that's really nice. Thank you very much.
Yeah. I have a problem with how not share my pulpit. And
okay, just 1 second. Maybe we can ask Aleksandra. She's the host of the meeting, or
can you simply do stop sharing?
I don't. Okay.
Yeah.
So, oh, thank you.
You're welcome.
Thank you, Aleksandra, and thank you, Dominika, again. So now we will have a presentation by Karolina Maciejewska from the University of Gdańsk. Unfortunately, she couldn't join us, but she shared a pre-recorded version of her presentation. So Aleksandra, could you help me with that and turn it on? Thanks.
Hello, my name is Karolina Maciejewska. I am PhD student at Department of Plant Ecology at the Faculty of Biology of the University of Gdańsk. Today I would like to present what I'm currently doing as a part of my doctoral thesis. The title of this presentation is Fossilized Plant Remains as a Source of Knowledge about the History of Meadow and Pastures Communities. The example from early medieval islands of the Lubusz land. Meadows and pastures are very important in economic and ecological terms. These plant communities are strongly associated with human activities like mowing and grazing. Meadows and pastures occur in various environmental conditions. Therefore, these plant formations are characterized by diversity in species composition. Many of plants and animals are rare and uncommon. Due to habitat fragmentation and cessation of traditional management, meadows and pastures are increasingly protected. There are numerous studies on the phytosociological changes and the floristic composition on contemporary meadows and pastures, but there is not enough information of their past. However, there are possibilities to get to know more about the history of these plant communities. Such data collection is possible by identification of fossil plant remains stored in various kinds of deposits. Important information can be obtained from the analysis of botanical materials collected at the archaeological sites. Archaeological sites are places where we can find preserved traces of ancient human activity. The science that studies plant remains in archaeological sites is called archaeobotany. Archaeobotany is a science of relationship between plant and human activity in the past, which based on botanical remains preserved at the place of former human existence. So in the various archaeological objects and cultural layers, we can find fossilized seeds, fruits, pollen, or vegetative organs. So, archaeobotany allows us to understand the relationship between human and plants on many levels. It is possible to obtain information on the plants' uses as food, medicines, or ritual items. On the other hand, human use the natural environment causes its changes. People in the past transformed natural plant communities into semi-natural and anthropogenic ones. Meadows and pastures were such kind of plant communities created by humans since the Neolithic. The use of archaeobotany and elements of modern cytosociology allow us to reconstruction the past of these plant formations. Now, after this long introduction, I would like to tell you about my research. A great example of excavation when the numerous plant remains representing meadows and pastures were preserved are early medieval artificial lake islands from the Lubusz land in eastern Poland. Lake islands are very interesting objects because they were created by local communities. Each of the islands had a wooden platform and a bridge connected them with the shore. Why they built it and how they use it? This is a good question. Their function is still not fully understood. However, these islands probably had economic, communication, and sacred function. These islands are very interesting for archaeobotanists. The material contains remains of over 100 plant species from meadows and pastures. Our material was represented by over 12,000 remains. The analysis of phytosociological affiliation showed that the identified plants referring to contemporary plant communities of 5 classes. This plant formation was different in botanical composition and structure. There exist in various soils with different humidity. So it was semi-natural and anthropogenic meadows and pastures on wetland on less wetland, dry grasslands, psalmophilous grasslands, heath and poor grasslands, and swamp meadows and transitional bogs. The land transformation for this type of plant communities began around 7th century with the entry of the local settlements. It seems that the people who used the free island explored the same plant formations. In the analyzed material, the greatest role is continued by species of wet and fresh grasslands, which, together with the significant share of species from other communities that tolerate water level fluctuation, give an image of strong economic exploration of area located in the vicinity of lake islands and related watercourses. Botanical traces also indicate a significant share of species characteristic to the dry grassland, although they were located rather in some distance from the lake themselves, but still within research of use by the island users. Due to limited time, I will focus on Molinio arenateretera and Nardocalunetea classes. So, vast majority of species currently belong to semi-natural and anthropogenic meadows and pastures of the Molinio arenateretera class. Grasslands of this class are particularly strong associated with human activities such as mowing and grazing. Many plants are of the greatest economic importance because they are edible by animals. The considerable taxonomic richness of this class allowed us to the identification of 4 orders within, which mean permanent or periodically wet communities, low grassland or periodically flood places, communities of fertile meadows on mineral soils, and communities of grasslands. Moreover, many of the identification plants could be used in variety of ways due to honey-giving, healing, and sacred properties. Plants inedible for animals may indicate wet infestation of this plant formation. Uninteresting in the context of meadow and pastures, as well as animal feed, are animal coprolites found during excavation. Coprolites are preserved animal fecals. The paleontological composition of goat and sheep coprolites found on the one island can be proof that animals were fed with plants from Molinio arenateretera class. The second significant class is heath and poor grasslands. They can form in a place of communities of the Molinio arenateretera class that have been improperly used or directly in forest areas. Heather, which means Calluna vulgaris, is a characteristic species from these plant formations. Flowers and seeds of this plant were especially numerous in Lubnivice, which suggests that this plant was deliberately brought to the island. Heather could have a sacral meaning and be used in rituals. This is a particularly interesting information due to potential sacral use of the islands. It is possible that this plant was an ornamental element of celebrated ceremonies. Another aspect of discussion plant material is the identification of fossil plant remains which, due to disappearing habitats and extremely diminished populations, are now under special protection. This is interesting information because the meadows and pastures communities in the Lubusland are subject to protection due to the cessation of extensive management, including traditional cattle grazing. Some were associated with Festucobrometea, which means dry grasslands, Chauceria sericeta nigra, which means swamp and meadows and traditional bogs, and Nardocalunetea, which means heath and poor grasslands. The continuation of research on material obtained from man-made lake islands brought new evidence of presence and role of local grassland in early medieval times in the Lubusland. It can be assumed that in the medieval times, these unique sites were constructed in neighborhood of well-developed meadows and pastures. It seems that people who used islands in Nowy Dworek, Chęcina and Lubniwice explored the same plant formations. The grasslands of the Lubusland were not only a source of fodder, but also a source of medicine and ornamental plants, as well as a place for grazing wild animals. At the end, I hope that you understand the role of archaeobotany in the reconstruction of past plant communities. My archaeobotanical analysis with elements of modern phytosociology led to describe meadows and pastures in the vicinity of early medieval lake islands. This subject is very interesting both for botanists as well as archaeologists. Thank you very much for attention.
Thank you very much. Do you have any comments or questions? I'm not sure if our expert on fossilized plants is here, but maybe.
Hello, I want to say I'm here.
Okay.
I choose the option of recording because I have too many problems with my internet connection and computer, and I didn't want to take a risk for something going to broke. So thank you very much for showing my record of my presentation. And
sure, thank
you
very
are
much.
Any questions?
And sorry for this miscommunication. I thought that you are not here, but yeah, everything went very smoothly. So very good idea. Do you have any comments? Okay, sorry for personal stories, but I have to say that it was really interesting for me because my background is hydrobiology, and previously I was working with freshwater ecosystems, so that was really interesting for me too. Okay, if we don't have any questions, thank you very much, Carolina.
Thank you.
And I think that to keep the momentum, we can move to our next speaker, Francesca Panissuti from the University of Milano Bicocca. And Francesca, you have a couple of extra minutes, so you can relax a little bit and take your time.
Okay, thank you. I'm now starting to share my presentation. So, okay. Oh, sorry, just a moment, sorry.
No worries, no worries.
Of course, there will be some problem.
If you prefer, we can share it for you.
Yes, I would have preferred to share mine, but actually my computer security is having some issues. So yes, I guess it is better if you share my presentation, because actually here I'm not able to quickly change the settings. Oh, maybe here we are. Okay, thank you. Sorry, I was all trying to change the settings. Okay, so I can start now. Thank you. Should I notify you when to change the slide? No? Okay, I'll start. So, hi to everyone. My name is Francesca Panissuti, and my presentation today regards the effects of mucilaginous bloom on Dicogonian forests of Tavolara Punta Coda Cavallo marine protected area. And in particular, it is focused on the involved species determination and Dicogonian mortality assessment. So during this, you can sorry, can you move to the next thing? Thank you. During this presentation, I'm going to start with a brief introduction on the main topics, and then I'm going to show you the materials and methods of this study and to explain to you the main results, concluding with a brief discussion. Gorgonians are important ecosystem engineers in the coralligenous habitat they shape. In fact, they are able to increase three-dimensionality and biodiversity. The four most important species are in the Mediterranean Sea are those in the pictures on the right: Unistalla cavolini, Unistalla singularis, Paramorisia clavata, and Unistalla verrucosa. These corals are as important as threatened. Firstly, by some of their intrinsic characteristics, such as the slow population dynamic, meaning delayed fecundity and low recruitment success. Moreover, they are threatened by some external factors, such as the current increase in seawater temperature due to climate change and the increase in frequency of harmful algal blooms. All these factors are leading to mass mortality events. HABs can be caused by macro or microalgae that can be pelagic or benthic. They can be also toxin producers or mucilage producers. In the second case, the bloom is characterized by the production of large amounts of mucilage made mainly of polysaccharides. The damages brought to the benthic ecosystems are that of direct suffocation of the sessile organisms and the creation of a situation of benthic anoxia with the same negative results. To these purposes, the aims of the study were to provide new insights and new knowledge on the effects of mucilaginous blooms on Gorgonian forests in the MPA. And in particular, the goals of the study were to assess the impacts of the bloom on two Gorgonian species, Unistalla cavolini and Paramorisia clavata, and to identify the algal species responsible for the bloom. This study was conducted in collaboration with the already mentioned NPA and in the framework of activities related to the Marine Strategy Directive and the Mediterranean, Interact Mediterranean Problem MPA ADAPT that is focused on the relationship between mass mortality events and climate change, and also in which the IUCN and ISPRA are involved. As you can imagine, the study was conducted in Tavolara Punta Coda Cavallo MPA. And here, two different substrata are present. Therefore, two sites were chosen, site 1 on limestone and site 2 on granite. Moreover, two transects were outlined per site. Firstly, a preliminary survey was conducted on both these already mentioned species regarding the abundance and health status of the colonies. And subsequently, it was chosen to further investigate only Paramorisia clavata. Sorry, can you go? Yes, also the next one. Thank you. So the first experiment regarded the mortality assessment and had as a response variable the branch percentage of different injuries. This was an experiment of image analysis conducted of both substrata. Here, 10 1-meter-squared photograms per transect were extracted from videos recorded in the MPA. And the percentage of branches belonging to 3 different injury categories was visually estimated and also estimated with the ImageA program as control. The 3 categories of damage were living tissue length, branches covered with epibionts, and denuded branches. The second experiment regarded the determination of the involved species, and in particular had as a response variable the number of cells. Sorry, can you slide? Thank you.
Sorry.
No, probably there is some delay in the presentation, sorry. This was, in this experiment, samples of mucilage and water were collected from Paramorisia clavata colonies present on the limestone substratum. Water was viewed under an inverted microscope through the use of sedimentation chambers, and also mucilage was viewed with slides as control. In fact, the most abundant species embedded in the mucilage are also those that most probably produced it. Of course, some data analysis was performed by means of univariate analysis of variance, and in case of significantly different results, and also an a posteriori SNK test was performed. Now I'm going to show you the main results of the study. And in particular, regarding the first experiment, as you can see here in the 4 plots representing the, 2 plots representing the 4 transects, the pink bars that represent the living tissue length are the largest. In fact, overall, the colonies were found to be for more than 50% healthy. Moreover, an interesting trend emerges looking at the other 2 bars. In particular, the yellow bars representing branches covered with epibionts are larger than the blue bars representing denuded branches. But actually, this result was not confirmed by the ANOVA. Moreover, interesting is the situation present on the limestone substratum. As you can see here, the colonies on T2 transects were found to be overall healthy. But important to mention is the fact that here a very small number of colonies was found. Regarding the second experiment, the bloom resulted formed by a benthic bacillariophyta, Cylindrotheca closterium. The genus Cylindrotheca is small but worldwide distributed in marine, freshwater, and brackish water. Moreover, the Cylindrotheca species is the only one of this genus that can be resuspended in the water column. And as you can see, I hope, it is characterized by 2 plastids pointed in blue and a central nucleus pointed in pink. Regarding the bloom, it was the 2 transects, no differences were highlighted in a number of cells. In fact, the bloom was proved to be uniform throughout this site. Sorry, it is in the next slide here. Thank you. Okay, so no differences between the 2 transects were highlighted. From all the collected data, next slide. From all the collected data, and regarding in particular the percentage of damaged branches per colonies, some differences were highlighted comparing substrata in relation to transects. And in particular, these differences can be found on the limestone substratum, where, as you can remember, there was a really small number of colonies on T2 transects. This could be due to the diving pressure, but actually this site is not a really frequented one. So maybe, next slide. So maybe some environmental conditions such as the strong currents present on T2 could be addressed as the reason for the low number of colonies. In fact, a very strong or discontinuous current can hamper the ability of the polyps to feed on suspended particles. Yes. No, sorry. Also another slide before. Oh no, so okay, it's okay. Okay, sorry.
Is it fine, or you need another slide? Sorry.
Yes, I need just more time here. I modified my presentation. Yeah. This one is the same. Okay, I just wanted to say a few other things about this experiment, meaning that another interesting result is the fact that the T1 transect was quite different from the granite transects. In fact, it was more damaged, and this could be addressed to the fact that these transects lie on a truly important and frequented dive site in the MPA. Moreover, the damage extension was found smaller than 50%, and according to literature, this result can mean that probably the colonies will be able to withstand epibionts competition and fully recover. Moreover, the absence of differences between the other 2 typologies of damage can signify that probably through the past years, the Gorgonian forests have undergone events of the same impact. So on the other hand, regarding the mucilaginous bloom, it was proven to be monospecific and caused by a diatom, meaning a microalgae. And this is quite important because it confirms some recent studies that underlie the primary importance of microalgae in blooms, but it's quite not in accordance with other studies that focuses in particular on the Tyrrhenian Sea, where the blooms were found caused by filamentous macroalgae, such as those in the pictures on the left, Trisonephos lewisi and Acinetospora crenita, for instance. But actually, on the other hand, other Cylindroteca clostridium dominated bloom have been analyzed in the Mediterranean Sea in the location that you can see in the map on the right. Concluding, this study can have some truly important practical application for the MPA that can be provided as management tools for the restoration and conservation of Gorgonian populations. First of all, some coral restoration activities can be performed, but actually this is quite an expensive practice. On the other hand, the punctual monitoring of the blooms through the years can provide knowledge useful for programming some physical action of mucilage removal that can be performed just for a few months per year during summer where the bloom occur, and just once or twice a month. So this is probably the most feasible solution. Thank you for your attention. And thank you for helping me with the presentation.
Sorry for not always being on time.
No, no, probably was the delay in the connection.
Thank you very much, Francesca. Now we have about 4 minutes for questions and comments. Okay, so I was wondering, at some point you mentioned that these blooms can cause anoxic conditions and hurt a bit Gorgonian forests. I was wondering if anoxia or hypoxia is a common problem in Mediterranean Sea, because I'm aware only of the problems in the northern part of the Adriatic Sea.
Exactly. Yes. Well, it is not common in the Tyrrhenian Sea, being it an oligotrophic sea compared to the Adriatic Sea, that is quite more shallow respect to the Tyrrhenian Sea. So no, it's not very common, but the accumulation of this huge amount of material and the fact that there are several bacteria embedded in it that start to decompose it creates this situation of anoxia. It's not common, it's a direct consequence of the benthic mucilaginous blooms.
I see, thank you very much. Yeah, we observe also anoxia and hypoxia in the Baltic Sea, and many scientific groups are interested in the consequences of that. I see that Com has a question. Com, please go ahead.
Yeah, that's a related question to the point you just had. I was wondering if you had any information on cases where the hypoxia around the Gorgonians also influence the, let's say, I wouldn't say the reef fish, man, the fish species that are kind of usually associated with these organisms. Do you notice that if the condition become very anoxic, do the fish assemblages changes and might go away or?
Okay, as far as I know, not really, because it is truly a situation related to the very bottom of the sea, and it affects mainly the sessile or not really movable organisms that cannot escape this situation. So like Gorgonians or maybe other organisms that cannot move. But as I know, fishes are not really impacted by the anoxia situation for now. Thank you.
Thank you. Do we have any more questions? If not, just let me summarize a little bit this session. Our speakers covered distinct views on different ecological processes. So we had these consequences of competition. We had also influences of the environmental conditions on the structure of the community, but also opposite perspective where community structure was used to reconstruct environmental conditions in the past. And finally, also, we were happy to see that native fauna is able to deal with some problems. So I think that that was a really interesting session, and all of our speakers deserve for a huge applause. So my proposal is to turn on all of the microphones now and give the applause to our speakers. They're really shy.
I can even show mine.
Thank you very much. Thank you to speakers again and all of the participants of this session. So
Thank
now
you very
we will have
much.
10 minutes of break and see you after that. Bye-bye.
Yes. Bye to everyone. Bye-bye. See you later.
No, actually, we have a lunch break now till 12:45 Central European time. So see you at 12:45.
Sorry for this confusion.
No problem.
Bye.
See you guys.
All right, so hello and good afternoon, everyone. This is Ahmed Rashad Jamalyar from Afghanistan, and your chairman for the next 60 minutes. First of all, it's my honor and privilege to be a part of this wonderful conference and chair the session. So in this session, we have 4 presentations by our expertise and researchers. Before we start the first presentation, I would like to remind my friends who are going to present their presentation to consider the following points. First, you will have 12 minutes to deliver your findings and 3 minutes for open discussions, questions, and comments. So definitely, I will let you know when you spend your 10 minutes of your presentation in order to be in safe side. So now I would like to kindly ask our first presenter, Ms. Nastaran Fedai Haidari, to present her presentation entitling In Search for Wisdom, Reconnecting with Literature as a Life Guidance for the Young. So Ms. Nastaran, the floor is yours, please.
Thank you, Ahmed. So I'm just going to try to share my screen, but if any error occurs, I'll just ask for your help.
Sure.
Thank you. So does everyone see my PowerPoint?
Yes.
Yes. Please
Okay,
go
great.
to full mode. Maybe.
Oh,
yeah.
sorry, just
Yeah,
a
it is.
second.
Yeah.
I need to go to the first slide first. All right, so hello, everyone. My name is Nastaran, and I'm so happy to be here today to attend the conference and to talk about In Search for Wisdom as a way of reconnecting with literature for a guide life, you know, to use it as a life guidance for the young, or maybe perhaps for everybody. Why only the young? And yeah, so, you know, many young people lack proper support or guidance around themselves. So, and in the process of decision making and choosing their life direction, they may risk getting involved in shady groups, sects, or even fall for extremist ideologies, which are really dangerous. And also, especially now during this global pandemic, many youngsters could not find their way out, unfortunately. And as a result, they got involved in drugs, truth or dare, and drinking games in order to ease the pressure that they are feeling. Maybe like, I don't know, it would be loneliness or some other struggles that they may have in their lives, especially during this global pandemic. And also we can see that the popularity of inspirational quotes and wisdom ideas prove that there is a great need for life guidance and wisdom exemplars, which are actually available for us in literature. And I would like to explain a little bit about mysticism to you. Some of you may know or may not know, but mysticism, especially from Persian mystics approach, means cognition. And it's a way of living, you know, it's like a lifestyle that you can choose to have. And cognition can have two aspects, quality and quantity. Quality is more about love and passion. Quantity is more about logic and ration. For example, what has benefit for me? What's in it for me when you want to choose something or do something? But from mystic approach, it's really important for us to have a combination of these two to be able to maintain a good life, because they are very necessary, I mean, as they complement each other and they are correlative. So today I have prepared 3 poems by Sohrab Sepehri, an Iranian poet and painter. I have also translated them into English. And so forgive my flaws if you see any in the translation of the poems. But before that, let me explain a little bit about Sohrab, who he was. Sohrab first started as a painter, and then he became a poet as well. At first, he was practicing classical Persian poetry, but then he got to know Nima Yushij, who was the founding father of modern poetry in Iran, and got inspired by him and continued his way. Sohrab uses short sentences in order to express his thoughts and emotions in a really beautiful way. He has references to mysticism, Sufism, Hindu mythology, Taoism, Confucianism, and Zen Buddhism in his poems. Also, unorthodox associations of words can be seen in his works. The first poem is called The Footsteps of Water. Life's a pleasant tradition. Life's wing is as vast as death. Life's a jump the size of love. Life's not something we put on the mantle of habit and forget. It does not matter where I am, the sky is always mine. Windows, ideas, air, love, earth, all mine. Why does it matter if sometimes the mushrooms of nostalgia grow? I don't understand why it said horse is a noble animal and nobody keeps a vulture in his cage. What's clover flower lacking before a red tulip? Our eyes need to be washed to see differently. Words need to be washed. A word needs to be the wind, the rain itself. We see that in this beautiful poem, Sohrab is pointing out lots of things. He talks about how lovely, how beautiful, how precious life is, and that we need to appreciate it, to respect it, and to cherish it. But of course, at the same time, he also talks about the other parts, like the downs of life, that, well, we all are mortals, we die eventually. And well, it's okay if sometimes we may feel nostalgic or down. We shouldn't forget that these moments will pass and eventually good moments will come back again to us. Also, he questions and criticizes conventional norms of the society. That's why some things are labeled as good or bad, as beautiful or ugly, as vice or virtue. And it's really important for us, you know, to have these things in our minds, to dare to ask, to question, and to seek answers to our questions instead of just blindfoldedly follow someone's path or someone's opinion. It's okay to get inspired by others, obviously, but it's also important that we would think and question independently. The next poem is called Umbrellas Must Be Closed. Let's close our umbrellas and go under the rain. The mind, the memory must be taken under the rain. We have to go with all people of the city in the rain. The friend must be taken under the rain. Love must be sought under the rain. You have to sleep with a woman in the rain. You have to play in the rain. Things have to be written, told, and planted in the rain. Life is soaking in the rain. Life is soaking in the pool of presence. Let's undo our clothes. Water is one step away. Sorry. So in this beautiful poem, Sohrab maybe kind of wants to encourage us to take audacious actions in life, to be bold, to be fearless, or to just sort of break free from all these traditions and norms that are given to us, you know, that are described for us to just follow as a way of living. I don't mean that they are bad necessarily, but it's also important that we also take our own actions instead of just, again, blindfoldedly follow something or someone or a path, I would say. And also, you know, you can see in this poem that he just kind of wants us to cherish whatever we have around us. Just to give rain as an example, when it rains, usually people complain. Let us be honest to one another. It's like, oh, it's raining again. And you may just open your umbrella or just put on your jacket and run home or just find a shelter for yourself. And instead of doing that, why not embracing the moment, embracing what is happening around us? And of course, rain is the example, but you could expand it to lots of things. It could be traffic, it could be red light, it could be, I don't know, like an unfortunate thing that may happen to us, lots of things. But if instead of getting annoyed or complaining or criticizing, if instead we try to embrace them, make peace with it, and accept it, we would, you know, just get out of these difficult times in a more, how to say, like in a better way, obviously, because I don't know, like criticizing life, getting annoyed and being sad is going to bring you down and eventually makes you sick or even depressed. But if you try to remain strong, make peace with it, you are going to be the ultimate winner. Same goes also about time, because if you try to fight with the time and you say, oh, why are you going so fast or why are you going so slow, then the time is going to go the way it goes. It's not going to change. The way we feel it is actually making us, how to say, think that, oh, it's going slow or it's going fast. But if we just try to go the same pace, the same flow with the time, then we make peace with it and we will enjoy it. Otherwise, we'll be the ultimate loser. And the final poem is called Let Us Not Mud the Water. Let us not mud the water. Down the stream, a dove appears to be drinking. Or in a forest far away, a sparrow is washing its wings. Or a jar is being filled in a village. Let us not mud the water. Maybe this running water goes by a poplar to wash away sorrow from one's heart. Maybe a dervish is dipping dry bread into the water. A beautiful woman a beautiful woman came by the stream. Let us not mud the water. The beauty has been doubled. What a tasty water, what a clear water. How pure the villagers are. May their springs run forever. May their cows milk well. I have not seen their village. Undoubtedly, there are footsteps of God under hedges. The moonlight there makes clear the breadth of words. Undoubtedly, in the upper village, the walls are short. Its people know what kind of a flower an anemone is. Undoubtedly, there blue is blue. A bud is blooming. The people of village are aware of it. What a village might it be? May its alleys and orchards be full of music. People at the head of the river understand the water. They didn't mud it. Let us not mud it either. Well, in this, you know, in this very beautiful and touching poem, we see lots of things. Well, there's a stream that is connecting all of us to one another.
You have 2 minutes.
Sorry?
You have 2 minutes.
All right. And we see that, you know, like the stream is just giving without asking. It's just feeding everyone, plants, animals, people, without asking who is benefiting from me, without having any expectations in return. Just like the sun is shining on all of us without asking who we are, who we worship, what color we are, how we look. So this also can be a very beautiful way of living if we take it as a lesson or as a role model for us to be kind, to be respectful, to be considerate, to be giving, but without having any expectations in return. this is good for ourselves as well, because when you help others, obviously you feel good about yourself. And when you don't have expectations in return, you are never going to be disappointed. And let us remember that this poem is kind of also showing us that we all are linked to one another, just like the stream that is, connecting us to each other. We may not see it, we may not notice it, but it doesn't mean that it doesn't exist. So it's good if we are going to be conscious about it and to have it in every action that we take, in every word that we say, or even every thought that we have in our mind, as we are going, as whatever we do is going to not only influence us, but the entire universe that is surrounding us. So as a result, we all know that humans by nature are always in search of universal truths and to find answers to their questions. And I hope these poems helped you, as I saw it, to see as well that these answers are within our reach. We just need to go to our literature, to all the messages that have been given to us and benefit from it. Thank you so much for your attention. If you have any question or comment, I'll be happy to receive them, and I'll do my best to answer as best as I can. You may also email me at nastaranfadeheydari@gmail.com. If you have any questions, I'll be happy to reply. Thank you.
All right. Thank you so much, and perfect timing. Do we have any questions from our audience?
Wait, sorry.
I think there's no question. May I ask a question, Ms. Nastaran?
Yes, I'm just trying to stop sharing.
Just stop the sharing and then.
Yeah, but I have reshare, not stop share. Oh yes, there it is. I guess I stopped it, right? Or no? Do you see my screen?
Still yes.
Oh. How about now?
Yeah, it's fine.
Okay, great, thank you.
Actually, I was wondering if he was, Sohrab was inspired, his work and influenced by any famous Persian poets, either in contemporary literature or in ancient time.
For sure, he was inspired by Nima Yushich, as I mentioned earlier, because Nima was the founding father of modern Persian poetry in Iran. And in the articles that I read about him, about Sohrab himself, it was mentioned that when he got contacted, by these poems and read them, he got really inspired and touched by them. And this is why he even had such a extreme shift, let's say, in his style, because at first he was practicing classical poetry, which has lots of rules to follow. It's very strict, that you have to have your verses in a certain way, you read them in a certain way, but then he suddenly just switched to modern poetry that has no specific structure, I would say, and like a poet's hands are open. They can just literally express their thoughts, their emotions, their feelings, their messages in whatsoever way they desire without having any restrictions or forms to follow or to obey, I would say. Yeah.
Thank you so much.
My pleasure.
Is there any comment, any question? Still, we have like 1 minute.
I think there's one question on the chat.
All right, let me track. Nastaran, can you please?
Yes. Robert is asking to what degree poetry is popular among young Iranians. Well, in general, honestly, poetry is very popular in Iran. This is also how we grew up, even in our important traditional, feasts or festivities that we have throughout the year. When people and families gather together, they read poetry at home. Usually elders like grandparents are reading for youngsters. And I believe it's still very popular, maybe not as popular as for like our grandparents' generation, but still I can see that many people around me enjoy reading poems, especially classical poems. And yeah, that's it. It is still popular, honestly. I can't say how popular, but it's still very popular.
Yes. Well, thank you so much, Ms. Nastaran, for the wonderful presentation. Wish you all the best. So let's move on to our next presenter. May I call Ms. Aleksandra Koroza to deliver her presentation.
I will share my screen.
All right, go ahead, please.
No, can you see it?
Yes, go ahead, please.
Okay. So I bet on it that most of you have heard about noise pollution or both traffic, if not both of them. But have you asked yourself actually how noise pollution might be really responded by marine animals, such as dolphins? So a good example here is how the threshold of hearing of humans and dolphins look like. So humans can hear from 20 hertz to 20 kilohertz, but 7 times higher are the frequencies which the dolphins can hear. Hi, I'm Alex, and I work for EOPAN, but today I will be presenting my master thesis from Bangor University in collaboration with Sea-Watch Foundation. So why is it exactly important to study boat traffic impact on cetaceans? First of all, cetaceans are top predators, meaning they regulate the whole environment. Their presence is crucial for well-functioning of it. And what is even more important is that they are really vulnerable to environment and they can be indicators of its health. So an example here is a noise pollution. We probably all have heard somewhere that cetaceans use sound on a daily basis for their hunts, socializing, and more activities. And same goes for the fame, which is increasing with the time for cetaceans. With that being said, we will need to manage well boat traffic. And this is where the marine traffic regulations should come in. So why I have chosen bottlenose dolphin as a species of my study? This is mainly because this species is really familiar for most of the society, but also because at the site of my study, this was the most abundant species and easy to watch. And now the increasing public interest in cetaceans has influenced the industry, more specifically the touristic one, where the dolphin boat watching industries are involving more and more. But also we have more access towards personal crafts, so more people can have it and afford it. With that being said, boat traffic is increasing even more. And that can have more consequences for dolphin behavior in short-term and long-term basis. So an example here could be the swimming speed of a dolphin. It can be observed as a normal or as fast. And for example, if it is fast next to the boat, this means it is a negative behavior, and it can furthermore be perceived as a long-term if observed on a daily basis for several years. And this can lead to further consequences for the health of the animal, as well as a health of the full pod dynamics. But observing behavior changes is not always easy, because there are some environmental factors which can stop us from doing it, such as sea state or visibility. And as I mentioned, my study was conducted in Newquay in Wales, in Cardigan Bay, which you can see on the left corner map. And now Newquay is a marine protected area, mainly because of the bottlenose dolphins, which are really important for the environment functioning. And they have there a lot of food and were a very good environment for nursing their calves. Now, because this is a protected area, there are several marine traffic rules to follow. So, for example, it is compliable with the marine traffic regulations to slow down in the presence of cetaceans and pass them slowly and stop to watch them, but it is not compliable to not slow down within 300 meters from them, as well as it is not compliable to follow them and change rapidly your course of direction or a speed. It is not really allowed to feed them or touch, but under the license that you can have, you can conduct research, which is also compliable with the code. Now, how exactly the data for this study was collected? It was collected between the 2006 and 2018 by Sea-Watch Foundation interns, including me, in 2-hour shifts. And every 15 minutes, we recorded environmental factors such as sea state or visibility, and more importantly, boat type, if they were present, and for example, visitor passenger boat. And then we written down its name, for example, analoids. And then if it was complying with the code I mentioned before, and if the animals, dolphins, were present, we noted their behavior and response towards the boat. There were several responses to report. So, for example, it could have been negative when dolphin was really escaping the boats. Positive, if it was really in a playful mode, let's say, and for example, bull riding. And neutral, if it was not changing its behavior. And if it comes to recorded behaviors, there were several, but some of the most important ones are socializing, when dolphins are rubbing, playing, and my favorite one is when they were playing with the jellyfish. And another behavior we recorded was swimming speed. So normal swimming speed or fast swimming speed. Also, we recorded resting behavior, which could have been observed when dolphin was near the surface and conducting shallow and short dives. Now, the most important results I got were for more frequently observed boat types. And those were speedboats, small motorboats, and visitor passenger boats. As you can see on the graph on the left, the negative response was more likely to appear than positive one. And for the boat names, it appeared that there was also a difference in dolphin response towards different names. So for example, there could have been a visitor passenger boats of different names that were responded in a different way. The next thing was that majority of the boats at Newquay were complying with the code of conduct. And as you can see on the graph on the left, dolphins responded to it positively. So the more there was a compliance in the code towards boats, the more the positive answer of the dolphins towards them. But still, there were some boats that were not really complying as much with this code. So for example, speedboats. And then we observed some changes of behaviors. For example, a decrease in arresting or suspected feeding in dolphins. I have done similar analysis for low frequently observed boat types. And thanks to that, I could observe that there was some adaptation towards known boats among dolphins. So the more frequently boats were observed, it was more likely for dolphin to respond towards it in a positive way. This is probably because they can learn how the boat behaves. What you could also see was the shift in performed behaviors of a key importance. And this was a sign of a long-term changes. Although not significant, those changes are important. Both parameters possibly have an impact on dolphin response, which you can observe by seeing the graph from my presentation of different response of dolphins towards different names of it. So
you have 2 minutes, Alex.
Okay. And animals affected by anthropogenic disturbance may remain at the site to perform important activities such as feeding or nursing their calves, like I mentioned before. So the solution is a good site management. As I said before, there is a code of conduct and marine traffic regulations at Newquay site. And thanks to that, majority of boats are complying with it and dolphin response is more positive. Now, in my presentation and observations, I haven't really included neutral responses of dolphins because I was looking at marginal behaviors and responses. But this site has a really good accomplishment thanks to those regulations included. If there is anything to improve, this would be to consider that one dolphin can be exhibited to several boat approaches because this is not really covered by the code of conduct at Newquay. And thanks, I think I have finished and thank you for your attention. If you have any questions, you're more than welcome to ask me now.
Thank you so much. It was really, really interesting, especially from my side, because my major is hearts, and that was really interesting. Thank you so much. So is there any question, comment, or any open idea regarding the topic from our audience side?
If I may?
Yeah, sure, please.
You said that they got accustomed to particular types of ships and they recognize them. Would that also go with the size of the ship? What I mean is, like, would they prefer the small motorboat or Titanic?
Oh, okay.
Size?
Yeah, so this is what was really covered by the boat name, which I haven't said. Maybe I should have mentioned it, because if you record the boat name, then you can see the parameters of it. And I didn't really go deeply into analysis of those parameters, but previous studies on this subject at this area did. And in fact, the boat size, length mattered, as well as the type of the engine. I think this might be because of maybe propeller type, different engines. And then the sound which is produced by the breaking bubbles of the water might be of different frequencies, but this would require, I think, some different type of analysis more further into this topic.
Sure, thank you.
Thank you. Any other question? Still, we have like more than 1 minute. All right. Thank you so much, Aleksandra, for the wonderful presentation. So let's move on and ask our third speaker, and I'm sure she's going to speak about effects of PVC microplastics contaminated soil on 2 wild plant species. Miss Laura?
Yes, I'm here. I'm going to share the screen, the presentation. Okay, can you see it?
It's about, yeah, go ahead, please.
Okay. Just a moment. I'm trying to. Oh, okay. Everyone see the presentation?
I think so.
Okay. Good afternoon, everyone. Thank you for giving me the opportunity to speak to the International Sopot Youth Conference of 2021. Today, I'm going to talk about the effects of polyvinyl chloride microplastic contaminated soil onto wild plant species. Almost 370 million tons of plastic were globally produced in 2019, and since the 1950s, there has been an annual increase in plastic production of about 8.6%. Parallel to its increase, plastic wastes are likely to increase as well, along with their accumulation in the environment, due to the persistence of these materials, together with the often ineffective waste management systems. Once they are released in the environment, plastics are fragmented into smaller pieces due to the combined action of microorganisms, weathering, and UV radiation from the sun. The smaller fragments that originate from these processes are known as microplastics. Microplastics are heterogeneous plastic fragments of less than 5 millimeters of different shapes and polymer type that consist of fragments, granules, and fibers that are considered emerging contaminants of concern and that can be found ubiquitously in all terrestrial compartments. Based on their origin, microplastics can be distinguished in primary and secondary microplastics, with primary microplastics directly produced with micrometer sizes as part of other products such as abrasive in cosmetics or drug vectors, and secondary microplastics formed by the fragmentation of larger plastic debris. In the last decades, research on microplastics has mainly focused on the presence and on the impacts of this contaminant in marine environments, where it has been observed that they can be ingested by various marine organisms, causing different effects, such as the blockage of the digestive tract or increase of a state of inflammation, decrease the growth, or cause liver stress. There are also evidences that suggest that microplastics are also widely present in freshwater environment as well. It has been estimated that 80% of marine plastic comes from terrestrial sources, and that there could be more plastic in soils than in the oceans. Despite this, there is a major knowledge gap on the presence and on the effects of microplastics in terrestrial environments, and in soils in particular. Microplastics may enter soils through multiple sources, including landfills and littering, the application of seawall sludge on agricultural fields, the use of wastewater for irrigation, plastic mulch films on agricultural soils, atmospheric deposition, and tire wear. In soil, plastics can be fragmented to generate micro and nanoplastics by biological processes of soil organisms, such as feeding or digestion and excretion. And moreover, the movement of these organisms combined with the physical characteristics of the soil can cause the vertical and horizontal migration of microplastics within the soil compartments. In addition to this, microplastics can also migrate to the surrounding environment through the action of wind, erosion, and surface runoff. Early research on microplastic pollution in soils suggests that there could be potentially negative impacts on soil biota, for instance, on earthworms, and changes in soil physical properties. However, there are little information available on the possible effects of microplastic on terrestrial plants, and most of the studies have investigated these effects on agricultural species. So the purpose of the present study is to investigate the effects of polyvinyl chloride, PVC, microplastic soil contamination on the growth of two wild plant species. We selected the native Centaurea chanus and the invasive Senecine equidens as study species. They both belong to the Asteracea family. Centaurea chanus is an annual species native to the Eastern Mediterranean coastal region, which has historically spread worldwide associated to cereal crops. And Senecio equidens is a perennial species native to South Africa, which has been introduced into Europe by the end of the 19th century with ship's hull commerce. It is currently invasive in many European countries where it can be found mostly in disturbed sites, such as roadsides, railways, and quarries. With a common garden experiment, we grown one individual of each species in a 1% PVC microplastic contaminated and non-contaminated soil, and each treatment was replicated 10 times for a total of 40 pots. This experiment was carried out for 45 days, during which we recorded plant phenology. In particular, we recorded the final the germination time and the emergence of the first and the second leaves. And by the end of the experiment, we measured some traits of the plants, in particular, the final height, the lateral spread, and the photosynthetic efficiency. Starting to discuss results about plants trait, we found for Centaurea canus individuals that plants in control pots were higher and were larger than those in treated ones. And in particular, PVC microplastics caused a decrease in height and lateral spread of about 21.4% and 10.4% respectively. A similar trend was found for individuals of Senecio equidens, where plants in control were once again higher and larger, and PVC treated soil caused a decrease in height and lateral spread of about 28.8% and 22.9%. We measure photosynthetic efficiency using a chlorophyll fluorimeter, which detects the chlorophyll fluorescence emission from a leaf sample. And this is a good indicator for photosynthesis in a plant. And we found for Centaurea canus that this trait differed significantly between treatments, and it was higher in control plants than in treated ones. However, this trait did not differ significantly between treatments for Senecio equidens plants. So it seems that the invasive species responded better to plastic contamination when considering photosynthetic efficiency. Regarding plant phenology for both plant species, we found that phenology was similar between treatments in the first phase of development. However, for both our study species, the second leaves emerged earlier in control plants than in those treated with microplastics. Microplastics can affect plant performance with both direct and indirect pathways. For instance, it has been reported that they can change soil physical properties, such as soil aggregation, water evaporation, or bulk density. And this can have a direct impact on microbial soil communities, whose structure and biodiversity highly influence plant performance and plant growth. Microplastics can also exert a toxic effect on plants directly from the residual monomers or additives that are released, or through the adsorption on the microplastic surface of chemicals such as heavy metals or persistent organic pollutants that are released in the soil compartment and can cause a toxic effects on plant. Indeed, our result shows that polyvinyl chloride microplastics affect plant growth for both our study species. And there are similar trends for other higher plants found in the literature. For instance, it has been reported that polylactic acid microplastics can inhibit the shoot and the root biomass of common bean, or polystyrene nanoplastics can inhibit the growth and can cause eco and genotoxicity in faba bean. However, almost all of the studies that are found in the literature mainly focus on agricultural species due to the potential risk associated to human health. And to our knowledge, this is one of the first studies that addresses this problem directly to wild, strictly terrestrial plants. In conclusion, recent research, and this study in particular, shows that microplastic can affect plants' performance differently based on microplastic size and polymer composition. In particular, in our study, we found that polyvinyl chloride microplastics can inhibit the growth and can affect photosynthetic efficiency of plants. However, we also found that different plants may respond differently to plastic contamination, and it seems that invasive Senecio equidens responded better to soil pollution than the native Centaurea chanus. To understand the mechanisms behind such effects was not in the purpose of this study. This is just a starting point for further analysis. I'm finished. Thank you for your attention. I will now stop the sharing. Okay. Oh, I forgot to
Thank you so much.
turn on the video. I'm sorry.
Yeah, thank you so much. So we have like 4 minutes. Do we have any question? Can we have any comment from our wonderful audience? No?
Okay.
All right.
Well, if
Thank you so much.
If anyone will have any questions in the future, they can write me and I will be glad to answer them.
Yeah, that would be nice.
Okay,
actually.
thank you very much.
Thank you so much. Thank you. So let's move on to our last presentation. I think this presentation is a pre-recorded, as far as I have received a message.
You want
Can
me to
we
share
have
it?
the pre-recorded? Yeah, please.
So good afternoon everyone, I'm Clarissa Raguso and in this presentation I'm going to report our last research focused on the detection of microplastics in Maldivian coral reef invertebrates. Coral reefs are one of the most productive ecosystems in the ocean and their importance is relevant not only because they support approximately 25% of all non-marine species, but also because they provide a variety of services to humans, sustaining the economy of more than 500 million of people. However, nowadays coral reefs are threatened by different stressors which can be either natural, such as global warming, hurricanes, algal overgrowth and diseases, or human-induced such as coral mining, sedimentation, overfishing and pollution. Regarding pollution, microplastics are the brand new emerging form of contamination and still very little is known about their potential impact on coral reef ecosystems. Focusing on corals, since they are suspension feeder, is very likely that they are exposed to microplastics. Evidence of microplastics ingestion by coral are solely coming from laboratory feeding trial experiments in which the ingestion rate could be potentially altered since the laboratory condition does not totally resemble the ones encountered in the environment. Under this view, it's really important to collect data on field. In order to monitor the abundance of microplastics in coral reef ecosystems, besides direct chemical analysis, it's useful to select marine organisms that may help in the survey. Sponges are sessile organisms playing a key functional role in coral reef ecosystems and capable to screen huge volume of water through suspension feeding. They are characterized by high filtration rate, allowing them to efficiently capture pollutants. However, sponges are still poorly exploited in this way. This study was conducted in the Maldives, only in the 7th largest coral system on the globe. The Republic of Maldives is an archipelagic state composed by 1,192 coralline islands, of which 187 are inhabited, 145 are resort islands, and the remaining one are dedicated to different activities such as plantation, agriculture, or are simply uninhabited. Even if Maldives represent a remote area, recent studies have demonstrated that microplastics may be found even here. At least 8 million tons of plastics end up in our ocean every year, and the amount in the surface water is well below the input rates. The Maldives could represent a potential sink of missing plastics as a result of its monsoonal regime or because of its vicinity to the Indian Ocean Gyre. Starting from this basis, the aim of this work was to preliminarily assess the presence of microplastics in Maldivian coral reef invertebrates, focusing on two different phyla, Cnidaria and Porifera. Particularly, we have validated and optimized two analytical methods for the detection of microplastics, combining optical and spectroscopical techniques. The detected particles were chemically characterized and quantitative analyses have been carried out. We work on one side on sclerotinian corals since they are among the foundation species that structurally build the coral reef ecosystems, and therefore is extremely important to study the environmental stressors, and on the other side on sponges because we believe they have strong potential for biomonitoring the microparticulate pollutants. In this slide, I reported the steps adopted for the microplastics analysis: 4 samplings, followed by sample preparation, particles detection, and statistical analysis. Starting from the coral sampling, a total of 22 individuals belonging to the species Sporites lutea, Pavona varians, and Posilopora verrucosa have been collected around the islands of Magudu in the Fafu Atoll. They were collected in 6 sites, characterized by 2 depths, shallow and deep, and 2 different exposures, inner and outer reef. Relatively to sponges, a total of 7 individuals belonging to the class Demosponges were collected in the water surrounding 2 different islands, the inhabited Magudu Island, the Fafu Atoll, and the resort island of Tutufushi in the South Ari Atoll. They were collected across 4 different sites, and on both the 2 islands a potential control location near an healthy coral reef and a potential degraded site near the rubbish dump in Magudu Islands and near the sewage pipe in Tutufushi Islands were selected. For what concerns the sample preparation, we employ and compare 2 different approaches: the physical extraction, based on the use of a dense salt solution, and the chemical extraction through the application of the pressurized solvent extraction. For what concerns the physical extraction, particles were counted and the limit of the size depending on the radiation length used for the chemical characterization, that in our case was the infrared. For what concerns the chemical extraction, mass measurements were determined using the mass spectrometry coupled with the pyrolysis gas chromatography. In this slide, I also reported the different steps, the main passages concerning the 2 approaches. For what concerns physical extraction, we had digestion, density separation, and filtration under vacuum. For what concerns the solvent extraction, we had neutralization, filtration, and the setup of 2-cycle extraction. For what concerns the particles detection, we rely both on morphological analysis using the stereo microscope and on the chemical characterization through the spectroscopical detection of the functional group of the plastic polymers. Particularly, we use an FTI hair microscope, which provided peak of absorbance uniquely related to the vibration, the frequency of vibration of the bonds linking the atom of the plastic material. In addition, the chemical extracts were submitted to the University of Pisa for the mass spectrometry determination after the application of the pyrolysis gas chromatography. Statistical analyses were performed using the one-way ANOVA for sponges, since they had the normal distribution, and the non-parametric Kruskal-Wallis and Mann-Whitney test for corals, in order to verify the presence or not of significant differences among the different species, size, depth, and exposure. Analysis on coral show a total of 128 particles. 40% of the total particles resulted to be microfibers that were confirmed to be mainly composed by cellulose by infrared spectroscopy. And since their amount was comparable with the background contamination encountered in the blank analysis, we prefer to do not further investigate them, focusing more on the other particles. Among the other particles, 13 resulted to be microplastics and 64 resulted to be mineral fragments mainly composed by the skeleton of the corals that probably escaped from the extraction procedure. Microplastic represented 10% of the total detected particles, and the most abundant shape resulted to be fragments, with a percentage equal to 38%. 23% of the particles of the microplastic resulted to be reddish, and vast majority of the polymers resulted to be polyethylene, with a percentage equal to 77%. followed by the 8% respectively of polyamide and polyester. Among the coral sample, maximum concentration encountered was 2 microplastic per gram, and the average concentration was 0.41 microplastic per gram. Microplastics were present in 2 out of 3 species, and the highest concentration was in Poecilopora verrucosa, with an average of 0.86 microplastic per gram. This higher concentration could be related to the branching morphology characterizing the species, which may allow a higher degree of particles entrapment with respect to the massive morphology characterizing the other 2 species. Microplastics were present in 3 out of 6 sites, and the highest concentration was encountered in Birofushi, with an average of 0.75 microplastic per gram. This higher concentration could be related to the vicinity of 2 islands commonly used as touristic destination and therefore more subject to the local anthropic pressure. However, comparing outer reef and inner reef coral, similar concentration of microplastic were found. This could suggest that probably pollution derived from the anthropic activities on islands and from the open ocean may play same important roles. Then, comparing deep and shallow corals, also in this case similar concentration have been found, and considering that the most abundant polymer encountered was the polyethylene, which is characterized by low density and should float in the surface water, we can assert that somehow it reached also the bottom, and this can be due to different factors such as biofouling, ingestion, or photooxidative degradation. Analysis on sponges show a total of 63 particles, among which 22 resulted to be microplastics and 28 mineral fragments mainly composed by calcite. Microplastics represented the 35% of the total detected particles, 91% of them were foams, and 68% resulted to be transparent gray, which was the most abundant coloring. Vast majority of the polymer resulted to be polypropylene and polyethylene with a percentage equal to 41, followed by the 9% respectively of polyamide and acrylate polymer. Among the spongy sample, maximum concentration encountered was 3 microplastic per gram, and the average concentration was 1 microplastic per gram. Also in this case, we look at the variability of the microplastic concentration according to 2 different variables, the islands and the sites. Similar concentrations were found on the sample collected from the 2 different islands, and this could suggest that probably pollution derived from inhabited and resort islands could have similar patterns. According to the different sites, microplastics were present in 2 out of 4 of them, and the highest concentration was present in the sample collected near the sewage pipe with an average of 2.5 microplastic per gram. In conclusion, we can say that we have validated and optimized 2 analytical methods for the detection of microplastics in demosponges and in stony corals. We have detected microplastic from various synthetic polymers with size range between 134 and 25 micrometer, and analysis showed the presence of microplastic both in corals and in sponges. As expected, we found a highest concentration of microplastic in sponges as a result of their filter feeding habits with respect to corals that are suspension feeder. No statistical differences were highlighted according to species, exposure, site, and depth. Implementation of clean room ISO 6 processes are underway to a better control of background contamination and enhancement of the limit of quantitation. Thank you very much for your attention.
Thank you. I think we have the speaker on board, so if we have any question, we may call her to respond. Ms. Barbara, can you hear us?
Yes. Can you hear me?
All right. Yeah. Do we have any question? Comment? Feedback? No? All right. Thank you so much. I think we have one message. Let me read it. Yeah. Okay. So it was just a kind of compliment. Wonderful
Thank you.
presentation. All right. Thank you, everyone, for your nice presentations, findings, and also sharing your thoughts. Honestly, it was a wonderful session, and I wish everyone best of luck for their further initiatives. So according to our agenda, we have 15 minutes break, coffee break, and then we will have our next session. So enjoy your coffee and see you back. Thank you.
Welcome everybody to this parallel A session of the afternoon. So we can start with the first talk given by Julian Guzowski from the University of Gdańsk with the title of Mg-nitroL-arginine methyl ester as a potential drug for Sanfilippo disease. Julian, the floor is yours.
Okay, so good afternoon, everyone. My name is Julian Guzowski, and the subject of this presentation is the autophagy introduction by ENEA as a potential drug for Sanfilippo disease. This work is a part of a bigger project financed by NCN. And Sanfilippo disease is one of mucopolysaccharidosis, a group of lysosomal storage disease characterized by accumulation of partially degraded glycosaminoglycans. It is classified as MPS type 3, so it is caused by 4 different genetic defects determining subtypes A, B, C, and D. In table, you can see the name of individual lacking enzyme in MPS 3 and the list of mutation which occurs in this disease. So main symptoms of lysosomal enzyme deficiency is accumulating glycosaminoglycans in lysosomes. In Sanfilippo disease, this glycosaminoglycan is heparan sulfate. And the major symptoms of Sanfilippo disease are cognitive and neurological defects. Among them, development delay, cognitive decline, lack of speech or speech delay, sleep disturbance, seizures, and hyperactivity and aggression-like behavior are the most commonly occurring abnormalities. Somatic symptoms also occur in MPS 3, however, they are less severe than in other types of mucopolysaccharidosis and appear very heterogeneous among patients. MPS 3 patients appear normal at birth, and the first symptoms are recognized usually at ages between 2 and 6 years. Their lifespan is an average between 2 and 3 decades, so some patients can survive longer. And autophagy. Since today there is a lot of ideas how to help people with Sanfilippo disease. I want to introduce you the autophagy and this is an evolutionary concept process which can be found in all eukaryotic organisms from yeast to humans. Autophagy is a process of degradation of macromolecules in the cytoplasms, particularly proteins of a long half-life, as well as whole organelles in eukaryotic cells. Lysosomes play crucial roles during this degradation. And in the picture, you can see particular parts of this process. First is isolation membrane formation, then identification and collection of cellular components for degradation. After that, there is a completion of autophagosome. Autophagosome goes to the fusion with lysosome and create autophagolysosome. And after that, it goes to the degradation of content with which is inside. Okay, and as you see, there is a lot of various activation pathways of autophagy. Also, there is a lot of activators of this process, but just some of them can reduce MPS-free symptoms. And in this case, the most important condition for autophagy activators is to cross the blood-brain barrier. And L-NAME is an example of molecule which can cross the blood barrier and also can activate the autophagy process. In this picture, you can see how it works, and I will try to explain it at this moment. So, nitric oxide, uh, a potent cellular messenger, inhibits autophagosome synthesis by inhibiting JNK1. It is an kinase, and thanks for that, and L-NAME is an inhibitor of nitric oxide synthesis. So thanks for that, L-NAME reduces the numbers of nitric oxide molecules and activate the autophagy process. And now I want to introduce you the aim of the study. And in this project, I tested the efficiency of L-NAME in heparan sulfate degradation in Sanfilippo disease. In this project, I used fibroblast cell culture. Cell comes from people who suffer from Sanfilippo disease, and I put it in a table in this table the characterization of these cells, and also in the picture you can see how these cells looks in a microscope image. Okay, and the first test in this work is the entity assay. The entity assay is a colorimetric assay for assessing cell metabolic activity. A yellow tetrazole is reduced to purple formazan in living cells. So I incubated cells in a microlith plate with different concentration of L-NAME, and it was made to prove that L-NAME is safe for the cells and safe for the cells. And after that, the next test was the dot blot assay. In this assay, I tested the level of heparan sulfate in the cells which were incubated in different concentration of L-NAME. I prepared the cell lysates and put them at PVDF membrane. After that, in the picture, you can see the next steps. Blocking the membrane, put the membrane into the primary antibody, secondary antibody, of course, substrate solution, and after that I had to develop the RTG film with this with the results, and it looks like at this image. So the research results. Firstly, results of entity assay. As you see, only the highest concentration of L-NAME reduced the level, but truly, according to these charts, I can say that L-NAME is safe for the cells, and the results of dot blot assay and in the picture you can see the image from RTG film. I scanned it into the computer, measured by a density program to measure the density of these points, and after that I prepared the these charts in which you can see that all of concentration of L-NAME reduce level of heparan sulfate in the cells in MPS 3C and also in MPS 3D. So my conclusion is that ENEA reduces the heparan sulfate level in the cells and also doesn't damage the cells, so it is safe for them. And moreover, in my opinion, this tested should be continued, and they can give us good they can give us the drug for Sanfilippo disease. Thank you for your attention. This is my reference.
Thanks to you. We have time for question. Are there any questions for the audience? No. If there are no one, I have one curiosity. When you track this kind of molecule for the Sanfilippo disease, you can reduce the symptoms of this disease in a sufficient way to normalize their life, or just is a just reduction to minimize them?
At the moment, it was only it was tested only at the cell. So I just tested that heparan sulfate are reduced only in cells. But you know, if in the next step where maybe we will go with animal model and try to put ENEA and test it how it works in animal model, and try to find out maybe how it goes with homeostatic and goes for a whole organism. There maybe I will I can explain I can yeah I can explain it, but at this moment I only tested how it works in how the heparan sulfate only I only tested the level of heparan sulfate.
Okay.
Thank you.
So, thank you very much. Are there any questions? Okay, if no, we can thank the speaker, Julian, again, and we can prepare and move to the next presentation, which will be given by Dominika Pankanin from the International Center for Cancer and Vaccine Science. And she's going to speak us about the role of ChIP-STAB1 pathways in mechanisms for neurodegradation.
Hello, everyone. Thank you, Julian, for a very nice presentation. Can you see my screen?
Yes, you have to switch for the full in the full presentation view mode.
Okay, I think it is visible now. So hello, everyone. My name is Dominika, and I am a PhD student at the International Center for
You are not in the full screen view of the presentation, in the presentation mode.
Okay. I changed it, but I guess it's not working.
Are you working with the double screen?
No, not at all. No. Maybe I'll share again. I hope that would fix the problem. Okay.
Okay, now you fixed perfectly.
Yes, good. Okay.
Yes.
So, hi everyone, I'm Dominika, and I am a PhD student at the International Center for Cancer Vaccine Science here in Gdańsk. And today I would like to talk to you about dementia and present my research results, which will hopefully bring us a step closer to finding a successful treatment for a broad range of dementia-related diseases. So the question I'm trying to answer is what causes neurodegeneration? I'm trying to understand the role of CHIP protein in neurons and its influence in dementia. So you may wonder why dementia? Well, dementia affects millions of people worldwide, being a cause of around 9 million deaths per year. The World Health Organization describes dementia as a syndrome occurring as a result of impairment of several higher cortical functions, which include memory, thinking, comprehension, calculation, learning, language, and judgment. These impairments are often seen alongside changes in emotional control, social behavior, or motivation. In this map, you can see the distribution of risk for dementia around the world. The mostly affected are parts shown in red. The highly developed countries are affected in the greatest extent. The lowest risk is present in Latin America and Caribbean. The number of people suffering from dementia is expected to rise each year, therefore making it one of the main focuses of research nowadays. Dementia can have many different forms. Alzheimer's disease is the most common form of dementia, and it is responsible for about 75% of cases, either on its own or with other forms of pathology. Dementia is a frequent complication of Parkinson's disease and often a late symptom. Studies show that a significant proportion of patients with Parkinson's disease has some form of cognitive impairment at the time of Parkinson's disease diagnosis. Huntington's disease is an autosomal dominant inherited disease that causes abnormal movements and coordination difficulties along with cognitive problems. It is a progressive condition that usually begins in middle age. Cognitive changes often occur early on, and dementia is a common feature in about 50% of people with advanced Huntington's disease. The nervous system is the main target of each neurodegenerative disease. It is composed of two major cell types, neurons and glial cells. Neurons communicate with other cells via the process of synaptic transmission, and this is done by sending electrical or chemical signals to each other. Nerve cells consist of cell body and two major types of projections which evict from it, axon and dendrites. Axon releases signal in the form of electrochemical waves from neuronal cell body, transforms it into neurotransmitters, and passes it to other neurons. Dendrites receive signal from other neurons and pass it to cell body. And appropriate functioning of neurons is necessary for efficient work of nervous system. Any disruptions in neuronal homeostasis may lead to its loss of structure, eventually resulting in loss of function. In such event, development of neurodegenerative diseases may be observed. As you can see here in this picture, not only neurons are affected, but also brain is affected, its structure. When we compare the healthy brain and a brain of a patient suffering from mild form of dementia and a severe form of dementia. Despite many studies have been done, the exact mechanism of development of neurodegenerative disorders remain unknown. My research is aimed to uncover molecular mechanisms which lead to neurodegeneration in Alzheimer's disease, Parkinson's disease, and Huntington's disease patients. So I'm trying to understand, do CHIP mutations cause dementia? And you may ask why CHIP. So CHIP is the main focus of my PhD work. And I think I have a very strong proof that CHIP is involved in neurodegeneration. And here I'll try to explain this to you. So CHIP is the carboxyl terminus of HSP70 interactin protein, and is a member of E3 ubiquitin ligase, functioning as a link between the chaperone and proteasome systems, playing a vital role in maintaining the protein homeostasis in the cytoplasm. Studies carried by COP group in 2017 showed that CHIP can bind to cellular membranes under conditions of acute stress, serving as proteostasis sensor. Once docked to the membrane, CHIP may alter protein homeostasis of the cell. This might then affect cell proliferation and survival. The hallmarks of age-related proteinopathies, such as Alzheimer's disease and Parkinson's disease, are misfolding and aggregation of proteins. These events are regulated by the complex and adaptive proteostasis network. So for regulation of protease, CHIP could potentially become a target for development of urgently needed therapies for neurodegeneration and age-dependent disorders. CHIP improves proteotoxicities in various proteinopathies. Both phosphorylated tau and beta amyloid interact with CHIP. This interaction further leads to ubiquitination and proteasomal degradation of these proteins. The disruption of this process may result in development of various neurodegenerative diseases. Other studies have shown that CHIP also targets Parkin-associated endothelin receptor-like receptor and alpha-synuclein for their proteasomal degradation. The association of CHIP loss of function mutations to neurodegeneration is a strong proof of concept of its involvement in the maintenance of neuronal health. However, we lack mechanistic insight into its neuroprotective actions. This is why a network-level view of signaling was needed to identify features of CHIP in neurodegenerative disorders. Our team initially used SHS-Y5Y cell lines to investigate differences in proteomics in the wild-type and the CHIP knockout models. The SHS-Y5Y cell lines were chosen due to relative ease and low cost of culture. Also, since SHS-Y5Y are human-derived, they express human-specific proteins. Our team performed twice independent liquid chromatography mass spectrometry analysis of a wild-type SHS-Y5Y and a CHIP knockout SHS-Y5Y cell models. The results obtained from this high-throughput study were confirmed by a low-throughput technique, Western blot, and next confocal microscopy was performed as another form of confirmation of previously obtained results. Unfortunately, today I cannot give you the names of the proteins that we detected, but I'll do my best to explain to you what is their role in the nervous system, well, I can't reveal the names since the data hasn't been published yet. So firstly, we determined if the steady state levels, the first proteome-wide detection and quantification showed that among the proteins which were mostly affected by loss of CHIP function, the two neuromodulatory peptides had the highest fold increases, and both also had significant p-value, presented in figure 4. As both of them have been involved in neuronal development and dendritogenesis, they were of particular interest, and therefore they were chosen for further validation. So we did Western blot, and we managed to confirm that increased steady state levels of protein 1 were expressed in a CHIP knockout, and this protein 1 has a mass of approximately 10 kilodaltons, whereas the protein 2 has a mass of around 160 kilodaltons. And these results were consistent with reports in the literature. The next step was investigation of protein 1 levels in the wild-type and the CHIP knockout SHS-Y5Y cells via immunofluorescence using confocal laser scanning microscope with a 6-degree immersion lens presented in figure 6. You can see our protein of interest visible in green. Immunofluorescence confirmed previously obtained results showing increased levels of protein 1 in SHS-Y5Y CHIP knockout cells in comparison to the wild-type cell line. The second protein-wide detection and quantification showed that among the proteins which were mostly affected by loss of CHIP function, even more neuromodulatory peptides had the highest fold increase, and both also had significant p-value presented in figure 7. And this is actually quite interesting because when I was analyzing the results with the help of a colleague who's a specialist in mass spectrometry, he asked me, are we looking for anything specific? Do you have any idea what you would like to see, any proteins that you would like to check? And my answer was not really. I just want to see what is the difference in protein expression in the presence and absence of CHIP. And turns out that all of the our mostly differing proteins between the cell lines, all of them are involved in the nervous system development. So this analysis confirmed that proteins 1 and 2 are upregulated in the SHS-Y5Y chip knockout cell lines. The third protein plays role in regulation of synaptic plasticity. Protein 4 takes part in nervous system development. Protein 5 is involved in neurofilament cytoskeleton organization as well as substantia nigra development. And this is where dopamine neurons of the brain originate from, forming nigrostriatal dopamine pathway, which is crucial in facilitation of movement. And one of the Parkinson's disease traits is a loss of dopamine neurons in the substantia nigra. Protein 6 is involved in cerebral cortex and hippocampus development, synapse maturation, regulation of axon diameter, and neuron projection morphogenesis. These findings suggest that CHIP does play an important role in neuronal development and homeostasis. The next step would be performing previously mentioned experiments again, but this time with use of cortical neurons derived from induced pluripotent stem cells obtained from Alzheimer's disease, Parkinson's disease, and Huntington's disease patients, as well as from healthy controls. We're expecting to see significant differences between the expression of neuromodulatory peptides depending on the extent of the CHIP mutations present in them, and therefore a disrupted development of dendrites in cortical neurons. The presented research plays an important role in the development of new treatment therapies and medications for neurodegenerative disorders, which are burden to millions of people around the world. A potential future preventive care could be developed in which newborn children could be screened for genetic predispositions for neurodegenerative disorders. This would then enable early introduction of preventive care to delay or eliminate the onset of disease in such patients. This study might be a crucial step in advancements in neurobiology and an asset to biology and medicine fields. And here are the references. Thank you very much for your attention. If you found this presentation interesting, please feel free to comment. And if you would like to ask any more questions, you can also email me. Here is my email address visible on the screen.
Thank you, Dominika, for this very nice and interesting presentation. Is there any question from the audience? Okay, if there is no one, I have one question for you.
Okay.
Related to the first slide that you show concerning the risk of that we have in the northern most industrialized countries.
Yes.
Related to the dementia. Okay, this risk is yet normalized for the life duration expectancy of the people with respect to the south part of the world, or with respect to Africa, for example, or is an expression of the different genomic characteristics of the people around the world?
I think the first one, I think it's standardized, I would say, but I was actually thinking it would be very interesting to, you know, use bioinformatics approach to take samples from patients suffering from dementia and check the CHIP mutation present in them, which one is the most common.
Okay, and coming to the end of your presentation, you showed another very interesting thing, that with this kind of approach, it should be possible to prevent it yes. With analysis from very young people, right? I understood well. So theoretically, you could do a DNA analysis and to prevent in the future the development, for example, of Parkinson and so on. This is the idea.
Mm-hmm.
Okay, So
yes, actually, the idea is that if we would know that chip affects neuronal growth, for example, and if there's a mutation present at chip, there is a very high chance and much greater risk of a child developing one of the dementia in the future. So then we could introduce preventive care even before even at the early age. And for example, I'm starting a new project now in collaboration with Professor Węgrzyn from Gdańsk University from the Department of Biology. And he studied this natural compound, genistein, which is proved to work positively and reverse the effect of Parkinson's disease. So I think it would be very interesting to check if, for example, we can use genistein to check if it could reverse the effect of a chip knockout in a neuron.
Okay, thank you very much. And due to the importance of this thing, I wish you a good success with your research. Okay, so if there are no other questions, we can thank Dominique again, and we can move to the next presentation. So that will be held by Christian Lukasic, and the title is quite interesting because it's the importance of being earnest, a student-produced radio play Act 1.
Yes, exactly. So, well, good afternoon, everyone. And let me introduce myself. I'm Martina Zdrojewska.
And I'm Christian Łukasik.
Yes, and we are Atheneum University students.
And we would like to share our experience with creating a radio play based on one of the Oscar Wilde's plays.
Yes. So,
but
Christian?
Wait a second, because I have to share the screen with presentation.
Yes, definitely.
So it will be fine. Okay.
Okay, good. So let it be. And here is the table of contents for our presentation. So as you can see on this slide, actually, we will discuss definitely on general, like radio play, the team we are working with, time and location, the process and benefits. So next slide, Christian, please. And like to start with, the importance of being earnest, or a trivial comedy for serious people, is a play by Oscar Wilde, and it is a farcical comedy in which the protagonists maintain fictitious personae to escape some burdensome social obligations. So, working within the social conventions of late Victorian London, the play's major themes are the triviality, with which it treats institutions as serious as marriage, and the resulting satire of Victorian ways, definitely. And as you can see on the right side of the slide, it is the first printed out version of the play. And next, speaking of the composition, and the play's popularity has meant it has been translated into many languages, though the homophonous pun in the title earnest, a masculine proper name, and the second meaning is earnest, the virtue of steadfastness and seriousness, poses a special problem for translators, definitely. And the earliest and longest handwritten drafts of the play labor over farcical incidents, broad puns, nonsense dialogue, and conventional comic turns. And one fun fact about the premiere: the play was first produced at St. James Theatre on Valentine's Day in 1895. And now, speaking of the critical reception, the light plot does not seem to tackle serious social and political issues. Though unsure of Wilde's seriousness as a dramatist, the critics recognize the play's cleverness, humor, and popularity with audiences. So, let it be introducing themes, first of all, triviality. And Wilde said himself that we should treat all trivial things in life very seriously, and all serious things of life with sincere and studied triviality. So the protagonist's duplicity, for example, in Algernon's Bambering and Worthing Double's Life and Jack on Ernest, is undertaken for more innocent purposes. It is largely to avoid unwelcome social obligations.
The play repeatedly mocks Victorian traditions, social customs, marriage, and pursuit of love in particular. In Victorian times, earnestness was considered to be the overriding value in society, originating in religious attempts to reform the lower classes throughout the century. A homosexual subtext is suggested in the play by many of the critics. It has been claimed that the use of the name Ernest may have been a homosexual in-joke. In 1892, 3 years before Wilde wrote the play, John Gambree Nicholson had published the book of pederastic poetry, Love in Earnest. The word earnest may also have been a code word for homosexuals, as in the following phrase, "Is he earnest?" The term bumbering used in the play is a stratagem used by people who need an excuse for avoiding social obligations in their daily life. The word bumbering first appears in Act 1 when Algernon explains that he invented a fictional friend, a chronic invalid named Bunbury, to have an excuse for getting out of events he does not wish to attend. The genre has been deeply debated by scholars and critics alike, who have placed the play within a wide variety of genres, ranging from parody to satire. In addition, Wilde's use of dialogue mocked the upper classes of Victorian England, giving the play a satirical tone.
Yes, definitely. And why we chose this? Firstly, it is a lightweight comedy, not too complicated to perform, presumably short, and plot is engaging and humorous. Moreover, some of us already have an experience with dubbing projects, and our love to voice acting was the biggest motivation to do so far.
Yes, I have experience in creating radio podcasts and music, which I gained during my studies about sound engineering, which helped a lot.
Yeah. Okay, let's discuss the A-team roles. So, male roles was the biggest obstacle, so we had to decide what character would fit each person the most. First of all, Christian. Due to Christian's experience with sound production and podcasts connected to mixing and mastering, so he was given definitely one of the main roles. Cornelia is able to produce a deep male voice, which could be also best for the main male role, and that's because of lack of men in the group, unfortunately. Paulina has also a nasal, acrid, deep, low, and somehow canting tone of voice, which fits best the role of the posh old Victorian lady. Carolina's jadedness, seriousness in case of tone of voice fits best the role of manservant. Julia, detached from emotions, calm, rhythmic way of speaking, English language somehow imposed the talking manner. Anya has a calm, soft voice, best for introducing. And next, Gwendolyn is a lady with flirtatious, jaunty talking manner with fancy, elegant, arc way of speaking. On the other hand, very steady. So I thought it suits me a lot. And next slide, extra tasks. Acting supervision was done also by myself because of, simply put, hobby. Being a graduate of the music school where I gained some acting experience was definitely helpful. Next is accent and phonetics generally. It is my huge hobby, and I must admit I have quite a wide knowledge in this area, gained mostly during English classes at university. And because of the previous experience with video and sound editing software, I was able to help Christian with the final production. Next, Cornelia, Carolina, Julia were obliged to find matching sound effects and background music. And last but not least, Anya was editing the script to make it look clearer and readable.
Classes were conducted via Zoom platform. In class, we were working on a script and editing it. We were watching a play. Inspiration we took mostly from the 1952 film adaptation. Recording, mixing, and mastering itself were all done during the time outside class at our friend's house. We spent approximately 1 month working on the project, just recording and mixing after initial preparations during classes online. Time spent in class oscillated between 3 and 4 hours. Practice was done outside class. We spent even up to 12 hours a day. Huge amount of work was done. Many takes were recorded for each character. Repetitions were necessary to achieve both phonetic and acting perfection. We preferred face-to-face meetings. Everybody's opinion was that the teamwork works easier this way. Also, recording sessions with professional equipment should not be done online. Finally, it was the project created in regards of the evaluation for the subject name language project done during 4th semester of MA studies. Let's talk about methodology. About microphone, it's a condenser microphone, great for musicians, broadcasting, and also voice actors. Pop filter was used to reduce popping sounds, plosives like PTK in English language, they are produced with aspiration. We did not have much time to record sound effects ourselves. We used YouTube Studio and YouTube Platform in general as a main sources, mostly because sound effects there are not copyrighted. Everyone liked the play and enjoyed working on it. Thanks to that, it went smoothly and pleasantly. We tried to do it in the most professional way as we possibly could.
Yes, thank you. And concerning benefits of what we have learned from this experience so far. First of all, mastering British accent, many lexical expressions, phrases were learned. Due to many takes and repetitions during recording, as well as to the phonology lessons given by me, whole group improved their pronunciation skills, intonation, and fluency.
We have learned how to use an advanced sound editing software like FL Studio 20 with its functionality, efficiency, and built-in library of sound effects. And moreover, our language command has improved, mostly by the method of shadowing parroting. It is the positive effect of constant repetitions. We put a huge effort into it, but on the other hand, it was all done in a fun and active way.
Yeah, and additionally, we also got to know more about older version of English language, mostly coming from Victorian times. Even slang expressions, contemporary manners, society issues, traditions were found, understood in a way of behaving among higher class people, known as savoir-vivre. The play shows hypocrisy of posh people, especially in Victorian area, which was very, very big and huge. We discovered what they do in the pastime, main occupations, and forms of entertainment. Yes.
Yes, thank you for listening. And to close our presentation, we would like to play the trailer of this play for you.
Yes, and before we started, I would like to give it the go to our project coordinator to have some words about it, about the project, because he's here.
Yes, thank you, Martina. Yes, I just wanted to say, because as the student's kind of teacher and kind of a facilitator, this project, this work which my students have done, has proven to be probably one of the best projects any of my students ever have done. And this project really consolidates quite comprehensively, like the amounts of work, academic work, language work, but also teamwork and like people skills, which they have been practicing and developing over the course of their study program. And the amount of work they've put into this project resulted in a beautiful radio play, which we will be happy to present on our Athenaeum radio in the near future. So please watch the Athenaeum website if you want to listen to Act 1 of Oscar Wilde's The Importance of Being Earnest. Thank you.
Thank you. So let me try to play this short 1 minute trailer, but I don't know if the sound everything from the technical
give it a go, Christian. Don't worry.
I'll give it.
Okay, I cannot hear anything.
Cannot hear? So it doesn't work, I guess.
You need to share sound. Actually, when you share something on the Zoom, there is a checkbox at the bottom and it says sound shared. So if you click it, then it should work.
Yes, I see it. Yeah, maybe let's try this again.
Yeah.
And I guess now it should work fine.
It's working.
Athenaeum English Language Department presents the radio play of The Importance of Being Earnest by Oscar Wilde. Act 1. Starring Cornelia Jończyk as Algernon Moncrief.
Tomorrow, Lane, I'm going bambering.
Christian Łukasik as Jack and Ernest Worthing. Oh, that's nonsensology. You never talk anything but nonsense. Paulina Grzybowska as Lady Bracknell.
Won't you come and sit here, Gwendolyn?
Martyna Zdrojewska as Gwendolyn Fairfax.
I'm always smart, am I not, Mr. Worthing?
Karolina Kucharska as Lane, manservant.
Yes, sir. 8 bottles and a pint.
Julia Bielecka as narrator.
Lady Bracknell sees Jack and bows to him with icy coldness.
Anna Fiałkowska as introducer.
The end of Act 1.
Coming soon on platforms YouTube, Spotify, and Athenaeum Radio.
Okay, so thank you very much for listening to us, and thank you for your attention as well.
Thank you.
Thank you very much for your project and your nice presentation. So I hope you a good success. Are there any questions or comments or suggestions? Okay. If no, I suppose you are all invited to the radio play to follow the Act 1 of the importance of being earnest.
Thank you.
So thanks to you. And this was the last presentation of this session. So I would like to thank again all the speakers for their presentation and their work. And we can close this one, which finished just 15 minutes before the session B. Then we will have the break and finally the poster session summary. So thanks again to all of you.
The link for the final session you can find in chat. I put it there a while ago. So if you're lost, it is there waiting for you.
Thank you.
Thank you very much.
Thank you very much. See you at the poster session.